Hello everyone, welcome to Ask a Scientist Gaming, where we combine mediocre gameplay with expert level science. I'm Ken Hansen, I'm an Associate Professor of Chemistry and Biochemistry at Florida State University. I'm an expert in light matter interactions, color chemistry, all things to do with light. But more importantly, joining me today is Professor Mike Roper. Mike, if you wouldn't mind telling us a little bit about yourself.
Yeah, sure. Hey, everyone. My name is Mike Roper. I'm a professor in the same department as Ken at Florida State University. And we deal with analytical chemistry. So we develop methods and assays and instrumentation. And one of our focus is on biology. So we're pretty interested in the biology of the pancreas and how glucose is regulated.
All right, equally important, what are we starting with? What game?
Tyson's Punch-Out. Tyson's Punch-Out.
Mike Tyson's Punch-Out it is. So if you wouldn't mind, Mike, let's pause for a moment and smile at the camera because this is the YouTube cover shot. Perfect. All right, game time. Let's do this.
Game on.
So Mike Tyson's Punch-Out, a 1987 release. This was one of the earliest boxing games. Turned into a pretty big franchise, actually. And Mike, when was the last time you played this aside from this week?
Oh, God. Early 90s, if anything, maybe late 80s.
All right. So it's been almost 3 decades, so we're going to see how this goes.
I can't believe that. So there are no cheats active right now.
Yeah, no cheats yet. We do have Game Genie codes backing us up here, but we'll start with that. All right. For those of you not familiar, this game has a pretty big difficulty curve. It starts out okay with Class Joe, and then it gets a lot harder really quick. All right, so we might as well start with the hard questions. Does anyone have any questions for Dr. Mike Roper? Again, he's an expert in lab on a chip, microfluidics, mass spec, analytical measurements, diabetes. Throw them in the chat if you have any questions.
Do what I can.
Nice. First knockdown.
First knockdown.
We're going to consider that a victory.
Against Glass Joe, I'm not so sure, but I'll take it.
All right, so we'll start with an easy one. So 6-year-old Mike Roper was not dreaming about being an analytical chemist, presumably. So what led you to where you are today?
Good question. I don't know for sure. We're always interested in science, pretty good in science and math, and my older brother, 2.5 years older. He was, when he went to college at UT Austin, he majored in math, and I felt like, well, I can't major in math, so I'll major in chemistry. So, went from there.
All right. Just kind of fell into it then, and then accidentally became a professor. Oh,
found I was pretty good at it, understood stuff, so I was like, just keep going with it.
I got to do a general disclaimer for the audience who hasn't seen Mike Tyson's Punch-Out before. If this was on Disney Plus, there would be a disclaimer saying there is stereotypes and mild racism in this game, with characters such as Soda Pop Ginzy and Piston Honda, and who else is there? I guess you guys will see. But yeah, heads up on that.
Yeah, we did not make the game.
Question from Bronco: what is microfluidics?
Microfluidics. So and yeah, the other question, MEMS are type of microfluidics, or I guess microfluidics is a type of MEMS. So in general, MEMS are microelectromechanical systems, and it's essentially MEMS is trying to take something like you'd find in the laboratory, a big instrument, and shrink it down to the size of maybe a credit card or something that perhaps you could like take out into the field and do measurements on or do measurements with, so that it's portable and all the electronics are integrated into this device. And so that combines kind of the electronics, it combines detection, combines sample preparation all on one device. And microfluidics is kind of a subset of that, where we use something again like the size of a credit card, and we etch channels in the glass, and we can move liquid around in very accurate and reproducible ways. And by doing that, we can, this is really hard to talk science and play the game.
That's part of the
fun. We can control the fluid, and essentially make really sensitive and fast measurements possible. Gosh, don't it.
So microfluidics, what the micro doesn't necessarily dictate size, right? It's a threshold of behavior, is it not?
So in fact, the original definition, they tried to say that one of the dimensions should be in the micrometer size range. And so some of the first ones that were built, the channel depths that we etch into the glass credit card size devices are maybe 10 to 50 microns deep. And therefore the volumes of the liquids that we move around are on the order of nanoliters, not smaller. So a nanoliter is a billionth of a liter. So very, 1 with 8 zeros in front of it. So very small amount of liquid has to be used.
AB, get up.
We got.
So there's also. Go ahead.
No, and then the great thing about it is when you go down to those really small dimensions, physics that would not normally make a difference at the macro scale, at the large scale, start to play a big role at these really small scales. And so we can take advantage of those and make our measurements really fast. And that's something that we try to do constantly in the lab, is try to make fast measurements. And
I was going to say, fluids on that scale, they behave very differently, right? Like you need T junctions to mix and Ys don't. And what's the craziest, I don't know, microfluidic phenomena, the non-Newtonian intuitive physics that happens?
Well, probably the thing that everybody, I don't know, you either fight it or you enjoy it, is the laminar flow that happens at fluids at these small scales. So in laminar flow, if two fluids come together, their fluid streamlines are essentially just straight paths. They don't mix very well. You can't get turbulent flow. You can't get really like tortuous flow paths or anything like that. So it's nothing like a river flowing or like the water in a culvert or something like that. The fluid behaves really nicely, and that's great in some respects. In other respects, if you have two fluids coming together, they'll just meet and then they'll just move right down the channel side by side, and so it's really hard to mix those two fluids. And so we have to allow diffusion to mix for the most part. There are some ways that we can take advantage of a couple other ways that we can try to mix, but mostly it's based on diffusion.
Von Kaiser
that can be a very slow process in the macro scale, but on the micro scale it can happen a lot faster.
All right, during the break here, what are you drinking?
This is a session ale, Founders All Day IPA.
I'm doing Proof 850.
What are you drinking?
Local Proof 850.
Proof.
All right, couple questions. Yes, ethanol is one of our emoticons, if you can see in the chat, Mike.
I see it, yes.
We got our emotes approved by Twitch. In their terms of service, they say, we are okay with drinking on stream, but we do not encourage excessive drinking. So, heads up on that. All right.
Good to know. Good to know. I don't encourage that either.
Bronco wants to know, where would you be bringing this where you couldn't have full instruments? Like lab on a chip, where would this be applied?
All right, so in my postdoc work, we were using these devices to try to speed up rape kit testing. So, there's this huge backlog of rape kits that have yet to be really analyzed. They just sit on the shelf because the methods, oh my goodness, the methods used to, oh man, I thought I had a star. The methods used to measure, like to try to do an extraction of DNA from, let's say, a swab, and then analyze that DNA to see who the perpetrator was. Take a long time. And so we were using these microfluidic devices and trying to speed up these processes because of the advantages of the microscale behavior. And so that's a big push right now. It's kind of in the clinical and forensics field to try to do really rapid testing. We try to make measurements that would normally take weeks happen in just a few hours or so.
That's a really good example. The goal is to reduce cost as well, presumably.
No doubt, yeah. The reagents that are used in those kind of assays are they're expensive, and so we can use a fraction of what conventional instrumentation uses, and so the cost is a fraction. And I don't know. There's probably the biggest maybe the biggest advertiser for our field, even though it's not a great example, is Theranos, which is, if you've heard of Theranos, you know it doesn't have the greatest publicity.
Yeah.
However, the technology that they're trying to do, where they were trying to measure disease markers in just a droplet of blood or so, that is absolutely happening. I mean, people are doing this.
Just they weren't doing it
in a legitimate way.
So they just oversold what they could deliver on, right? And they Ponzi schemed it.
I don't know all the
degree.
details of the Theranos, but yeah, I think they just essentially over-delivered. But people are doing this, and there's a lot of examples of new companies starting up that are taking microfluidics and making assays really fast. And things, you know, you're probably starting to find more and more point-of-care testing. So that's like your blood glucose monitor or a home pregnancy test is the most common point of care. So it's a test essentially that any layperson, non-scientist could use and get really accurate results. So microfluidics is a big part of that, and other types of technology that we're interested in. Ooh, oh, come on.
Nice. Oh, so close.
I have a blister on my thumbs, I'm pretty sure, by the end of the night.
Yeah, and the rectangular controller digging into your palm. Flashbacks, man. It's a good kind of suffering. All right. Bronco wants to know, how's an analytical chem lab different from other chem/bio labs?
So, yeah, I would say, I mean, I think it really depends. That's kind of there's all different types of chemistry, and, you know, I bet you Ken's lab and my lab are probably pretty similar with instrumentation, certain types of instrumentation. Oh, goodness. Knocked out. Oh, paying attention. But we do a lot of measurement techniques, so we build our own. Maybe something that we differ, I don't know, but we build a lot of our own instruments. So we'll build kind of the optics, and then we'll build the electronics to measure from those optical systems. So it looks a lot different than like I don't even think, maybe we have some Eppendorf flasks in the lab, or some I don't think we have any kind of Erlenmeyer or anything like that, but we have a lot of probably looks more like an engineering type of a lab sometimes. There's multimeters and lasers and optics and things like this.
Yeah, I think I'd adding on top of that, and Mikey, you can correct me, but I think the accuracy with which you guys want to measure things is typically much higher than most of our labs.
Well, yeah, when we shoot for something like 5% RSD, you know, so we're looking to make a measurement and make sure that it's correct, right? So we don't want to report a number that's off by very much. Oh, man.
Well, so you would hate reviewing in my field, because we have something called hero devices, where if your solar cell behaves well once, that's the number you report in the paper. and sometimes it gets through without critique and anything. So
What?
yeah, no error bars, just hero device. That's the one you publish.
That's the classic question I get to ask almost every other student
Yeah.
during their oral exams or something. Where are your error bars on those?
I should know. You were on my first student's committee, and you did ask her that. Rightfully so.
Is that right?
Yeah. We don't publish hero devices. She had actual error bars. We just don't usually graph them.
Okay,
But yeah.
that's good.
All right.
The best is when they have the error bars and they're too small to be seen. That's like, oh, you can
Yeah.
shut the professor up.
Yeah, they turn a circle into a square because the edges come out. Yes.
That's right.
That's perfect. All right, QZeno wants to know, with such small instruments, even smaller sample injections, especially biological samples, how big of a problem is sample homogeneity?
Come on. That's a great question. Somebody's a scientist on here. Somebody's a ringer. Sample homogeneity is a huge problem. And that's something that we struggle with. We try not to bias our measurements or bias our results by looking at a really homogeneous sample. Sorry, I should clarify that. Biology is extremely heterogeneous. So if you're only looking at this, if you kind of bias, you can bias your results by just choosing what kind of biology to look at. Let's say the cells that we're interested in, we'll take the smallest ones, or if we take the most round ones, then all of a sudden our results are going to be biased because it's not the real population heterogeneity. So that's something that we struggle with. We're trying to make devices and make enough measurements that we can really truly understand the heterogeneity in a population and understand where it comes from and why would it be important to have that kind of heterogeneity. Great question.
All right, case 987.
It's a problem, but it's also something that to be looked at, actually.
So case 9876 wants to know, in your opinion, what is the most interesting application of microfluidics?
My own work, of course.
Your own work. I like it.
No, I'm kidding.
I mean, trees use
so there are some people
microfluidics,
who
right?
are building instruments to be put on like the Mars rovers and sent out into space and do analyses of extraterrestrial sort of samples and things like that, trying to do analysis on Mars and out in space. I find that fascinating. We're getting more and more into research that I lost to this guy before, didn't I? I remember his goofy grin. We're doing more and more analyses of like the biology people are developing for with stem cells, trying to make designer organs to try to help try to replace the islets and type 1 diabetes and things like this. Hang on. So I find that really fascinating, those biologists who can do that. I mean, that's super fascinating. And then people are making mimics of the blood vasculature in these devices, and then the cells sit in there inside the blood vessels inside your in vitro device, and you can do drug screens that mimic what's actually happening in vivo, in your body. So I think those are really cool applications. Oh, shoot.
Does a plant or a tree getting water up to the top, does that count as microfluidics?
Sure.
All right.
I'll allow it.
I don't actually know the size of the pathways, but it's like capillary action combined with respiration and things like that.
I'm not a microfluidic snob.
You're not a fluidics elitist? Speaking of microfluidics, a fun factoid for everyone. So when I start the stream, I have the stream title, but I also have a notification that gets sent out to everyone. I cannot put microfluidics in my notification. Something about Twitch's filter doesn't allow me to put microfluidics. I can put micro, I can put fluidics, but not together as one word. That's flagged some kind of swear word things. The only thing I could think is somehow it's an iteration of microdicks, but I don't know for sure. So that's my fun factoid for the night.
That's a great fun fact.
Yeah, I tried. It wouldn't let me update if I kept the word microfluidics in there. So that was not part of the notifications for this stream. All right. Calm Down Bronco wants to know, how do glucose monitors work?
So probably the most common ones are electrochemical. So that's the ones where you'd make a pinprick, right? You pinprick and then you put the blood on a little card, and the blood seeps up into, there's an electrode. There's 3 electrodes, and it measures the glucose in your blood. There's a couple of other glucose monitors that are coming online, and I guess they're trying to be made more and more, or less and less invasive. So there's some that, like, I think goes on a watch now. So it's supposed to be able to read the glucose level through your skin on your watch. That's an optical-based method, I believe. And then I think that's the, I don't know. I don't know a whole lot about glucose, but the most common, the blood one, is an electrochemical detection. They use actually an enzyme to convert the glucose into something that can be measured electrochemically.
And this is it. Round 3. A little bit of health left.
Oh, look at this. I can't beat that.
That's a tough one.
You have any hints on this? What to do?
I think you'd have to block it. I don't think you can move out of the way. Hey, Rob, yes.
Again.
So Alex Rob designed our emotes, and now he has it. I don't know if you saw it in chat. Thank you, Alex. Appreciate it. All right. Do you want a game, Genie?
Yeah, because I've hit the same guys, right? I've gotten this guy several times. I can beat this guy.
So if you lose in a fight, you go back 1 round and fight that guy again.
I see.
So
how about,
you guys can kind of see the list of Game Genie codes showing up on the bottom.
Oh, can y'all see that?
Yeah, yep.
No. All of a sudden, I'm going to get a lot better really quick. Watch. Should I do all of them or what?
You might as well. It'll let you march through most of the guys. All right. During our brief intermission, for those of you just joining us, joining me today is Dr. Mike Roper. He's an expert. He's an analytical chemist that studies studies and develops lab-on-a-chip technology, including microfluidics, and does things like mass spec to try to understand cell signaling, in particular with a focus on diabetes and understanding that. Oodles of Omics. That is an awesome name.
Oh my goodness.
If you are running a lab on a woo, I don't know what that is. Thanks for doing this, both of you. Found this on Reddit. I was wondering, for your lab on a chip experiments, what are your normalization strategies if you want to perform proteomics, metabolomics, and transcriptomics? What are your normalization strategies?
So you do realize I'm playing a game right now, right?
That is the point, Mike. Mediocre gameplay plus expert science.
I see.
You can only let us down on one of the 2 things.
So normalization of our methods. So we don't do a lot of proteomics. We just published a paper where we did our first LCMS project, and so I guess the question, like, I'm not sure what the question exactly is asking, but our normalization strategies, one way to do it is we'll take cells.
Pause before going into details. What is a normalization strategy? What does that mean?
So I guess the way I'm thinking about it is, if we, that's what I was going to explain. If we put cells on our device, and we're trying to see what they're releasing in time. That's what we're trying to do. So we try to keep our cells alive, and so we'll take the cells from the pancreas that release the different hormones that control glucose level. They're called islets of Langerhans. So we'll take these islets and we'll put them on a microfluidic device, and then we'll try to keep them alive. And we'll try to see what they're releasing in time, the different hormones. So if we want to be sure that, let's say, a big islet is, if we're counting it the same as a smaller islet, we would try to normalize that. We try to say, well, the big islet secreted or released like 3 times as much as the small islet, but is that really fair? Because maybe its volume is 3 times as big also. So we try to normalize based on, we'll take the islet out of the device and then we'll measure its total hormone content. And so we normalize to the total hormone content of the cells. To give you a very scientific answer. I think that's probably how you would do a lot of those other ones too. Metabolomics too, I guess you would normalize to either protein levels in the cells or DNA amounts. Transcriptomics, I don't know. I've never done transcriptomics. I'm trying to do a collaboration actually with someone at our College of Medicine who's an expert in transcriptomics. So we're trying to kind of get into that field. She's into it more than we are, so I can't answer that part though. Did that answer the question, Ken?
Yeah, that's good. Yeah, also shout out to Reddit. The number of subreddits that just cater to, niche expertise. Like there's a microfluidics subreddit, there's a lab on a chip subreddit. Just anything you want to learn about, there is an environment for you to do that. So Oodles, thank you for joining us from Reddit.
That's a great name. I love it. Yeah.
Oodles of Omics. On point. Well done.
Systems biology was taken.
I also like your name though, Mike. Microfluidics or Mufluidics. Pretty good. That's what I should have done. The U-fluidics. That would get past the sensors.
That would have gotten past. These are also called UTAS, so microTAS, so micro total analytical systems, or lab on a chip. And
I mean, that's
the first paper that came out on that was like in the late 80s, I think, maybe late 80s. There's this guy named Andreas Mons, and he just kind of talked about the concept. It wasn't even like an experimental paper. It was just like, well, what if we did this? And then he started work. I guess then the next paper that, like one of the 2 years later or something, they came out with a microfluidic chip doing something. And so it was just kind of a cool thing. He was like, what if we miniaturized everything, put it onto a device, and that would be this micro total analytical system.
So I'm curious, relating back to the other question about proteomics and metabolomics. So you have a tool set, a measurement skill. So when you go to conference, you have, a lab on a chip conference, but they're studying very different things. I mean, is there a language barrier between what you're studying and what they're studying?
It's a good question. There can be, but I think if you're working a little bit in biology, like we do bioanalytical. That's kind of how I feel like my subspecialty is, bioanalytical chemistry. I feel like I could listen to most bioanalytical talks and get an idea of at least the problem, maybe the significance of the work, and maybe even the innovation of it. And I guess the same can be said for lab on a chip sort of things. If people are presenting, even if they're talking about the Mars Explorer, if they explain their science well enough and like what they're doing on their science, I feel like it's okay. I can still get it.
So it really depends on who's presenting. Also, congratulations. Your Rocky 2 moment, you got your revenge, took the win.
Finally did. Took me all the cheats.
No, we count those. Those count as a win. And then you get the training montage. Is everyone ready for this?
Oh, look at that. Fantastic. Got my pink bunny suit on.
your world-class
Shout out to
boxer. You can
Christmas
wear
store.
anything you want. All right. Mike has defeated Piston Honda. Calm Down Bronco wants to know, what kind of cell signaling mechanisms do you look at?
So we, all right, so you're gonna take me down a path, I guess. So if you give glucose to one islet, Langerhans, like I told you about, the insulin that comes out of it, we see like these oscillations. The insulin kind of fluctuates in these pulses. And so we're really interested in what causes those pulses of insulin to be output. Son of a... Hey, wait a second. So we're really interested to see what makes these pulses of insulin appear. And then it's even more interesting, so we're really curious because if you measure insulin in the blood levels right outside the pancreas, those same pulses appear. Somehow all the islets in the pancreas are releasing their insulin or at least the majority of them, are releasing their insulin at the same time in these pulses. So how in the world can they do that? What's causing them to synchronize? That's something that we've been looking at for a long time. So those kind of cell signal mechanisms. So what makes them pulsatile, and how could they synchronize together?
Sorry to interrupt, Mike. Can you turn your volume down just a little bit?
Sure.
Sorry, we're doing a remote feed.
Okay.
So we've thrown a lot of words around here, diabetes and insulin and glucose, and I mean for the general audience, just walk us through the relationship between those things. Like, what is diabetes?
Right, okay.
How does it relate to glucose and insulin?
Yeah, good question. There's a couple different types of diabetes. The two main types are type 1 and type 2 diabetes. Type 1 diabetes is thought to be an autoimmune disease. So your own body destroys the cells in the pancreas that produce insulin. And if you don't have insulin, your body doesn't know what to do with the glucose. You can't get rid of the glucose in your blood, and that can be a very dangerous thing. Because in the long term, glucose is quite toxic to your cells. So you need the insulin to lower glucose levels in your blood. So in type 1 diabetes, destroys those cells that produce the insulin, and therefore you can't get rid of the glucose. In type 2 diabetes, it's much more, I shouldn't say it's more complicated. It's a different disease, but the overall problem is still the same. You still have too high of blood glucose. The mechanism, though, is quite different. There's partly your cells that should be responding to insulin are not, and partly the cells that are releasing the insulin are not working properly. So you kind of have this kind of dual problem. But again, the net result is that you have these elevated blood glucose levels.
All right, so on that explanation, we're going to throw our first prediction out there. For those of you that are interested in spending some imaginary internet points, we're going to throw up a prediction right now. It's going to ask which form of diabetes is more common, type 1 or type 2. Again, remember, everyone is on the Ask a Scientist honor code. You cannot Google. You just have to use your intuition or previous knowledge to make a prediction. Spend your internet points. You can find them in the bottom left of your chat. It's the Xi symbol. We call them standard internet units because they're entirely made up. But it's your opportunity to bet on which one's more common, type 1 diabetes or type 2 diabetes. And if you're not following us, click follow right now and you get 300 free internet points to spend as you choose. You also help get our visibility and numbers up. So yes, feel free to click follow right now and get your points and spend them on betting on type 1 versus type 2 diabetes. That may be your brother in chat. Is that?
That may be my brother. Got Moon Patrol, got Mario Brothers, and we do have track and field actually. I told you we use the spin. And let me borrow also the pad to actually try to play with the kids. I haven't had a chance to do that yet.
The power pad. I should have made that as some kind of betting thing.
Good Lord, can you imagine us doing that?
Oh, man. I'll tell you what, when we do Ask a Scientist in person, we will do that.
we need to have the dance.
Yeah.
The dance off.
Yeah. If somebody donates 1,000 bits, we will do that.
How in the world do you beat this guy?
Oh, man. This is a very lopsided prediction right now.
500 as he says he falls off of it. Yeah, no kidding. So have we been used for viral tests? No, we haven't really been using. We haven't been doing too much with COVID. There's a lot of competition in that field right now, and oh come on, that's not true.
All right, so Mike, just sorry to interrupt. Which is the more common type of diabetes, type 1 or type 2?
Type 2 by a huge margin.
That is what chat also decided. So 4 people to 0 voted type 2. Yes, it is type 2. I think according to, what was it, the CDC, it's 95% of people with diabetes have type 2.
Yeah.
But the number's reversed if it's children, right? It's type 1's more common?
Something like for that. Sure. I mean, type 1 is also known as like juvenile onset or child onset. Type 2 is adult onset. But it's turning out that that's kind of flipping a little bit too. More and more children are developing type 2 because of, well, playing video games and sitting around the house and not eating properly and things like that. So
that's complete bullshit. We did all that. No, it's probably true. It's worse now because of, touch screens and whatnot. All right, Oodles of Omics wants to know, do you see increases in insulin response to artificial sweeteners? Have you looked into artificial sweeteners?
I have not personally. I don't think they cause insulin to be released. These artificial sweeteners, I don't think they're super good for you either, though. But I do not believe they have the same mechanism for causing insulin to be output. So, they may have a low glycemic load. They may not cause insulin to be output very much, but I don't think that means that they're super healthy either.
Oodles, thank you for the tier 1 subscription. We really appreciate it. So subscriptions to our channel basically pay for beer money as well as Uber rides when people are drinking in person at my house. But yeah, we really appreciate the support. We don't necessarily need it. If you guys are going to subscribe, attach your Amazon Prime account to your Twitch account. Subscribe through Twitch Prime. That's essentially a check written to us directly from Jeff Bezos. You don't have to spend anything. So yeah, use your Amazon Prime to subscribe to us or anyone else. Again, it's free subscriptions laying around basically Amazon trying to support their own sub program. All right, so yes, Oodles of Omics, thank you very much for the subscription. Power glove for punch out. Scuzzbot wants you to use the power glove. The thing is, I could have given you a power glove to play punch out with.
What's a power glove?
Oh, you don't remember the power glove? It's you put it on your hand and it had a. Here, I'm going to show everyone.
Couldn't take it. He had to actually show people the power glove. It's gonna be like Thanos.
It is so bad.
Do you mean it's so rad, or do you mean it's so like...
That is the power.
Oh, look at that. Sweet.
Yeah, so it has infrared sensors in the front, and you put a bar across your TV, and you could play Nintendo with this. So it has the controllers and inputs here, and then, yeah. So the line is, it's so bad is from the movie The Wizard, which was basically a 2-hour Nintendo commercial. It is absolutely amazing. It does not work at all. But again, in person, when we do this again, you will try the power glove for everyone.
Oh, I'm totally trying the power bar. Whatever it's power glove.
Power glove, yes.
Power glove.
It was kind of a failed experiment by Nintendo. All right. Going back, catching up on a few questions. Sorry, the Legend of Tingle. Subscribe with Prime. Thank you, Legend of Tingle, for the subscription. Again, use your Amazon Prime. Write a check from Jeff Bezos directly to us. He has money to spare. All right. Going back to Scuzzbot 10's question. Will there ever be a cure for diabetes?
Oof. So this could be a prediction one. It's hard for me to say, but type 2, if you'd eat properly and exercise well, then that's a pretty good. You can avoid it. Easier said than done, right, these days. But technically, you could avoid it. So really, it comes down to type 1, and even if you have type 2, there's some pretty good therapeutics out there right now, like metformin and things like this, to help you control your glucose levels. Type 1, I think there's some really interesting work. This is kind of what I mentioned earlier. Like, there's some really smart biologists who take your, let's say, your stem cells, or can take certain cells from your body and convert them into stem cells, which can then turn them into they can turn them into the cells that make insulin. And so even if you have this autoimmune disease, so that your own body's attacking and killing your beta cells, you would have another source of beta cells. So even if they die, you'd have more of them to be able to replace them with. And so that's something that people are working on right now, and a lot of progress has been made in the last 10 years, and who knows what's going to happen in the next 10. So I think that's a very promising therapy for type 1 diabetes. And then there's other ways of making kind of these cells that release insulin to replace the ones that are killed in type 1 diabetes, besides just using stem cells. So those are definitely potential therapeutic options for type 1 diabetes. But you will probably always have this autoimmune response. So even if you inject new cells, you may have the same problem happening. Your body will try to attack those same cells. And I think there's work trying to be done understanding like why the immune response is doing this, and trying to stop that. But I don't. That's starting to get outside my field a lot. So I don't want to make it sound like I'm a clinician or an immunologist.
With that said, we'll put you on the spot. How optimistic are you there will be a cure for diabetes?
Type 1?
Mm-hmm.
I definitely think in my kids' lifetimes, for sure.
Awesome. All right, to calm down Bronco, you kind of addressed this a little bit earlier. How does diet affect type 2 diabetes?
A lot. There's if you gain too much weight, you can become insulin resistant. So you essentially, if you put on too much weight, then your body won't respond to the insulin that's being produced. And so the cells that make the insulin will start outputting more to overcome that insulin resistance. And you could live that way, or some people then develop even further, and pretty soon your cells will just stop making the insulin. They'll just be worn out, and so that leads to like overt type 2 diabetes. So certainly weight has a big factor in that. Certainly there's a lot of studies looking at like carbohydrates, and if you're ingesting simple sugars and things like this, that you're just outputting a lot of insulin all the time.
So is there I'm not familiar with this, is there a genetic component to type 2?
Yeah, definitely there are some genetic factors associated with type 2 diabetes. So you can be more prone to type 2 diabetes.
Tim21, thank you for the follow. We definitely appreciate the follows. All right. The rural juror Shout out to Unbreakable Kimmy Schmidt on Rural Juror. What are advantages of glass microchips versus PDMS? How easy is it to manufacture chips in PDMS versus glass? So break those words down for an average viewer.
So those
30 Rock is Rural
30
Juror.
Rock.
Sorry. Yeah, my mistake. I'm an idiot. Anyway, sorry. Question. PDMS versus
right.
glass.
The glass and PDMS that I was being asked about, those are two different types of materials that microfluidic devices can be made in. So we either make them in glass or plastic, typically are the two main types of materials that you make microfluidic devices in. And then there's a couple different types of plastic. The most common one is this thing called PDMS. The glass is pretty nasty to make. We use hydrofluoric acid to do the glass etching, which it's nasty stuff. And I still remember, I think there was a warning label that came from Sigma, or it was like on their MSDS, their material safety and data sheet for the HF. It said that HF will strip the proton off your grandma. I don't know why that stuck with me for a long time now.
That's not even how HF works, though, right? It's
I don't know. Yeah,
it's
just it's true.
That's amazing.
I just remember seeing that on the sheet from Sigma.
Yeah.
So we don't like making them, but they have some really good qualities. It's a lot easier to detect in glass than it is in some of the plastics. So we get nice optical detection in glass devices, but they're a lot worse to fabricate in. PDMS is pretty easy to fabricate. You can make several devices in a day, whereas glass usually takes maybe 2 days to make a couple.
So what's the disadvantage of PDMS then?
Good question. So it's almost like contact lens-like material. So you can think of like it's an elastomer. It's kind of a pliable plastic, and there are some molecules that are kind of absorb into the PDMS, and so it can be hard to work with in that regard.
Don Flamenco down. Congratulations. There's a link to hydrofluoric acid. My favorite factoid I learned about your lab is your fume hoods had to be equipped with hydrofluoric acid safe exhaust, right? Or like walls, something like that.
Don't even get me started. This is a whole thing.
That's fine. We got time.
Yeah, so we moved buildings, 2 years ago? We moved from one chemistry building to a different chemistry building. And we have a special type of fume hood, chemical fume hood, that when we're working with HF, we usually work in there. And in the old building, that was hooked up to like the normal exhaust fans that all chemistry buildings have, essentially. When we switched buildings, these engineers came over and they said that we needed special fans. They all needed to be plastic and all this stuff, and so they essentially had to like install like new ventilation into the building, and which I don't think is necessarily true, but I wasn't paying for it, so I was like, all right, go ahead, yeah.
Yeah, HF. Yeah, my favorite scene from Breaking Bad and the realistic depiction of acid or HF's application to materials is when you put it in a bathtub, it'll actually etch away the ceramic material. So HF eats away glasses of any kind, but it will not etch away plastics. And so that's why you keep HF in a plastic bottle. And so it's a weak acid. It's not as strong as HCl or HI or HBr, but it's more damaging to tissue, to glasses, all sorts of things. So yeah, it's kind of a terrible thing to work with. I guess if you get it on your skin, it's small enough it infiltrates through and starts triggering the muscle contractions. So it feels like you're running a marathon despite not doing anything. So yeah, pretty...
I think calcium fluoride too has a pretty low KSP, and so it'll suck all the calcium out of your bones, and I think it also destroys your nerves right away, and so you don't feel it on you. there was a story when we were in. I got my PhD at the University of Florida, and when we were starting to learn to use HF there, we had to go down to their kind of their clean room and their engineering, you know, get all in the bunny suit and all that stuff. And the guy we were working with, he had, I guess, had gotten some acid underneath his watch when he was working with it at one point in his life. And he said he got home and he took his watch off and there's this big burned part of his skin because he didn't feel the HF on him. So, pretty nasty.
That kind of relates to Bronco's story. Any lab horror stories?
Yeah, just one. I kind of wonder who this Bronco guy is.
He's one of my former Gen Chem students, and he's one of our regulars. Bronco, thank you for all your questions.
Oh, is that right? Okay, good.
So I have a couple. One is I have 7 stitches in my right index finger from an NMR tube. If you're not familiar with NMR tubes, it's basically a really long, thin tube of glass, typically quartz. And I broke the end off of it, and it shot up through my finger, and I had 7 stitches because of that. So that's the most damaging experience I've had in lab personally.
Wow, yeah.
Yeah, no, the NMR tubes are very fickle. Hairline cracks, and they just shatter in your hand.
Yep.
Oh, Scuzzbot Wotus has requested take a drink. Maybe you can wait until you have a break against King Hippo. No pressure.
Is that his name, King Hippo? Yeah, you think so.
Yep. Oh, Bronco has also redeemed take a drink.
Yeah, I gotta good because I'm out, so I need to pour myself one real quick.
No worries. Yeah, stack them. We're gonna go against Twitch's terms of service. We have 2 requests to take a drink.
Okay, fine.
All right. Cheers, everyone. Thank you for joining us on Wednesday night to talk science and play video games and drink alcohol. So, thank you all.
We, when I was getting my PhD, we had a, you know what piranha is, right, Ken?
Cheers,
you
Mike.
know piranha solution?
Yeah, it's the HCl plus nitric acid.
So I guess the way we made it up was sulfuric and
sulfuric
nitric, or sulfuric and hydrogen peroxide. That's what it is. And so we would try to neutralize it by like titrating in some sodium hydroxide when we were done with it.
I don't want to get technical on you, but you didn't take a drink.
No, but I accidentally poured in like a very high concentration of sodium hydroxide, thinking that it was much lower concentration. So I added a lot of volume to the piranha,
yep.
And it sounded like a bomb went off in the field. It just went boom, and luckily the sash was down, and I looked down and I had all this liquid on me. And I had HF in the hood at the time, and I was like, oh my gosh, you know, is this the HF? I didn't really know what was going on because I didn't know what I had done wrong. And I realized that I had put up the HF, so that wasn't HF. Mm-hmm. And I went and jumped in one of the lab showers just in case, and stripped down essentially, and my friend went and got me some clothes. So that was not a fun day.
That is amazing.
I was okay. It was just kind of destroyed the shirt that I was wearing.
I have never had to use the lab shower. You might be the first one I've known personally that actually had to do it. All right, Rural Juror, thank you for the follow. Mike, you owe them 2 drinks. There's been 2 requests for drinks. So cheers, Mike. Thank you for coming on Ask a Scientist Gaming. We appreciate your insights as well as your Mike Tyson gaming skills. A few questions we got to catch up on. Legend of Tingle wants to know, has there been advances in checking blood sugar levels to where you don't have to constantly prick your finger? I think you mentioned one briefly earlier.
Sorry, can you repeat that again?
Have there been advances in checking blood sugar levels to where you don't have to constantly prick your finger?
Yeah, in fact, there's some constant glucose monitors right now where I think the glucose monitor just sits, essentially it gets, I don't know the correct word, kind of attached to you or implanted. I don't know if it's implantable. I don't know exactly how it works, but it can continuously monitor your glucose levels in your body, so you don't have to constantly prick. And then these are really cool for like, they want to do like closed loop systems so that it's constantly measuring your glucose and then can deliver the right amount of insulin at, you kind of let the algorithm, the computer algorithm, figure out how much insulin, instead of you making a measurement of how much glucose and then trying to figure out how much insulin you should inject in you. So that's like a closed loop system. That's kind of the holy grail of what people want for type 1 diabetes.
Oh, Bronco,
that's what we were using Piranha for. We were cleaning our glass microfluidic chips before bonding. We have to bond the two pieces of glass together. So we clean them and then bring up the temperature to like 650 Celsius, and then the two pieces of glass essentially fuse together to make our device.
Yeah, fun factoid on cleaning glassware. Typically you don't want to use KOH solution to clean volumetric flasks because it actually eats away a layer of the glass and changes the volume of the container. Whereas I think Piranha's okay, right? That's just terminating the ends with hydroxides.
Silica, we do the same thing with some of our we use capillaries, fused silica capillaries. So if you use base, you'll strip off a layer of
Yep, and you change the volume and all sorts of things, yeah. Oh, Bronco, one follow-up on the lab horror story. Speaking of fume hoods and exhaust vents. So in undergrad, I was working in a lab. We were doing like thiol capping of quantum dots. And so the building was old and it had an exhaust system where essentially the exhaust from one hood went to the intake of the building. And so we were working with thiols, and thiols smell like rotten eggs, but they also smell like a gas leak. Because like if you have a methane gas leak, methane doesn't have a smell, so they dope it with thiols. And so that's where the gas smell comes from. It's from thiols. And so they actually evacuated the building, had the fire department come in, and only to figure out it was just a fume hood and the wind blowing in the right direction to put that thiol into the building circulation system. So yeah, I evacuated a building as an undergrad.
I bet you're proud of that.
I look back on it fondly because I didn't do anything wrong. Like, I handled everything correctly. It was just the building architecture and the wind that day just made it troublesome. So,
that sounds like that.
Oodles of Omic wants to know.
What is this guy doing?
Block. If you block like 4 times in a row, he'll stop and bobble his head. Block, bike. Just press
good
down.
Lord!
Just press down, down, only down.
He said block. I'm trying to. It's the controller, right, Ken?
It might be. Those controllers are a little bit jank.
Just like your architecture of the fume hood and all that. Yeah, it wasn't my fault.
Yeah, totally not. Eugene asked, "Did you try blocking?"
No, I hadn't thought of that. It's a great question.
Do you want to guess who totally not Eugene is?
Yeah, let me guess.
All right. Oodles of Omics wants to know, favorite paper you read in the past year?
Ooh. Oh, damn it. This. Favorite paper in the last year. Well, we just wrote a review paper where we had to read a bunch of papers. There's some really cool science going on. There was a paper that came out from kind of a combined group of Florida and Miami, I think, that was looking at small molecules being released from islets, and they were showing that these small molecules can be pulsatile also. So this kind of matches up with our insulin study, so that's a really cool paper to read.
So I understand some of those words. What is that in layman?
So islets of Langerhans, the ones that release all the different hormones, they also will release small molecules and potentially talk to the nerve cells that are penetrating the pancreas. And so they found that these kind of neurotransmitters that are released from the pancreas, they have dynamics, or they have oscillations just like just like their insulin ones. So Spot, what is, have you tried getting good? That wouldn't be any fun.
Oh, you get a rematch.
Yeah, so they had the same kind of oscillations that I was talking about earlier with the insulin oscillations. Every 5 minutes, they were seeing these kind of neurotransmitters also being pulsatile. So that was really cool to see.
Sorry to interrupt, Mike. I'm going to give you a hint on this guy. When he ducks down to the left or right to do a punch, when he squats down, do a low punch to his gut.
Okay.
So like on the right, do a right punch to his gut.
Sure.
Up, up, down, down, A, B start. Konami code. I don't think that applies to Tyson. We're already cheating a lot with Game Genie. These cheat codes are useless, says Bronco. There you go. And just alternate. Right and left. Nice.
Oh, nice. Okay.
All right. 9 o'clock. Anyone just joining us? This is Professor Mike Roper. He's an expert in lab on a chip, analytical chemistry, quantifying things accurately and efficiently, mass spec, microfluidics, diabetes, anything you guys want to know about those topics, or life in general. Mike is happy to share his opinion on anything.
May not like it.
That's not why we're here. We're not here to make friends. We're here to impart knowledge. So if anyone has questions for Dr. Mike Roper, please feel free to throw them in chat and we'll be happy to answer them. Blocks.
4, 5. Oh, I did 4 in a row. I counted.
Okay, it's like 5. Omic wants to know a very specific company question. Thermo, Waters, or Agilent guy?
Oodles of Omics, do you work for any of those? Or are you like trying to...
Yeah, do we need a disclaimer here? This video is sponsored by Oodles of Omics.
They're all great companies. I enjoy all of their instruments.
I love when they send me samples for free.
That's right. Right now, our mass spec that we're using a lot is the Thermo. So the Waters, though, I mean, they're well known for some awesome instrumentation, and Agilent is a beast.
So Agilent sells one of the most popular UV-Vis spectrometers on the planet, the HP8453. It's like the go-to default detector array UV-Vis. Orbi traps for the win.
That's true. That's from Thermo.
Oh man, Mike,
Come on, that was like a bunch in a row. We gotta skip, I think, just
2,
to Tyson.
3.
That's an hour of Tyson punch out.
All right, we're gonna skip right to Tyson.
Let's do it.
Let's do it. So you want to do it with Game Genie codes or without Game Genie codes first?
Is there any point to do it without Genie codes?
Yes, because we're gonna have a bet whether you can last 30 seconds with Mike Tyson or not.
Alright.
All right, so we have a code we can skip directly to Mike Tyson. And Mike Tyson is the hardest fight in the game, so you guys are gonna see us skip directly there. So we're gonna turn off all the Game Genie codes and we're gonna do a prediction.
Whoops. So I saw one question come up. What's our most useful instrument in the lab? Do you know, Ken? Do you know the most used analytical instrument in the world?
I'm gonna say UV-Vis. No?
pH meter.
pH meter. Oh, that's something we take for granted.
See, everybody takes the pH meter for granted.
Yeah. It's a non-trivial
useful
thing.
instrument in our lab, probably a.
All right, Mike, before you start, Prediction is
Before I
up.
start what?
So we're going to give people 2 minutes to place a bet. So will Mike Roper last against Mike Tyson for greater than 30 seconds or less than 30 seconds? We're going to go believers versus skeptics. Bet your standard internet units now. Let's gamble. Is Mike going to last less than 30 seconds or greater than 30 seconds? No Game Genie codes versus Mike Tyson, the final boss of Mike Tyson's Punch-Out.
Should we tell them what I did earlier today? I mean,
No.
just to brag a little bit.
Nope. We're going to see what people bet on. You use your standard internet units wisely. Will Mike last less than 30 seconds or greater than 30 seconds?
Yeah.
If anyone's not following, click the follow button, and you'll get your 300 internet points. You can bet on Mike Tyson Punch-Out. The code is 007373.
Five.
5963. But don't press start yet because we still have people betting.
Okay. I'll just keep going like this so it doesn't turn off.
Yeah, it's Nintendo. You never know. All right, get your bets
We
in
do
now.
use the Raspberry Pi. Gotta believe, baby.
All right, we got 10 seconds ish. All right, go ahead and hit it. 6-3. So we got 5 non-believers at 95% of the betting, 1 believer at greater than 30 seconds, Mike. Here's your chance to prove the skeptics wrong.
Excellent.
We have 1 believer. Do it last 30 seconds against Mike Tyson with no Game Genie codes. These are valuable internet points.
They've seen me play already and they know that this ain't good. Oh, son of a...
For those of you that
I'm
don't
back
know,
up. I'm
Mike
back
helped
up.
with
I'm
betting.
back up.
Mike Tyson is a 1-punch knockout.
What? 21 seconds.
All right.
All I need is 10 more. 10 more seconds, baby. Dodge, dodge. Uh-oh.
There it is. 40 seconds.
Boom! 48.
He did it.
That's a world record
Believers,
right there.
take the win at 48 seconds. Mike Roper lasted 48 seconds with Mike Tyson with no Game Genie codes. Those are not normal human seconds. Yes, those are video game seconds. You want a refund on your standard internet units? All right. So, greater than 30 seconds. Congratulations, believers. Do you want to put the Game Genie codes on and try it again?
Yeah, do this.
Man, what was the payout to that on the Skeptic?
Thank you, Eugene.
Wow, 21-1 payout on that the believer. Well done. And Bronco wants to celebrate by taking a drink. Mike, let's take a drink.
Well, get all these cheats in first.
48 seconds. Well done. Eugene will never bet again.
Well, Eugene, if you don't bet against me, you don't have to worry about that.
Your skepticism is what cost you there.
Yeah.
Believers
that's your
take
fault.
the win.
It's a 3, right?
Yep, 373.
Let's see how bad my short-term memory is. 373.
5963. Man, a 21-1 payout. That's the biggest I've seen. Mike, you earned them their internet points. Well done.
I don't know if that's an insult, kind of, a little bit. Is that
You know, we're skeptical. All right, so now we got Game Genie codes. You basically can't take damage.
if I could do a Tyson voice, I would totally do it.
Omic says, this is why I went to school and quit my professional poker career. Wow. So Bronco bet 50 points to get 1,000 back on that bet.
Wow.
Yeah, 20-1. Oh, Make no mistake, Mike Tyson is one of the hardest boss fights in video gaming.
Man, it's almost impossible, right? Like,
almost impossible.
people actually beat him?
I'll give you the next fun factoid. So there's a GDQ is a fundraising event where people speedrun video games. So the record for the entire game of Mike Tyson's Punch-Out is about 15 minutes. But there was a run at GDQ like 5 years ago where two of the best players played it blindfolded. So they played the entire game blindfolded, but they both shared a controller, blindfolded, and beat Mike Tyson.
So it was just one hitting like A and the other one hitting B? I mean, how does that work?
So one's controlling A and B, the other one's controlling the direction pad. And they played it together, blindfolded, and beat it. It's an amazing clip. If anyone's seen it, I recommend it. Or if
they beat Tyson too?
Yeah, they beat everyone. I mean, so there's like sound cues and there's certain manipulation you can do. It's not completely random. It's actually.
Sure.
Oh, there it is. Scuzzbot put a link in the chat if you guys want to watch the the blindfolded Mike Tyson fight. But don't do it now. We're paying attention to Mike Roper.
I was going to say, let's just do it now. It's probably more entertaining than this, to be honest.
I mean, all right.
Look at him. I got him on the ropes, baby. Get back.
Right, so during the intervention or intervening time there, you answered, what is useful instrument in your lab? What was your answer? Is it pH meter?
What is my answer? Gosh. Oh, I got a star. Mass balance or pH meter doesn't count, then I would say we do a lot of fluorescence, so we make a lot of use of like a plate reader in the department,
So
which measures the fluorescence of our samples.
So that's your most useful instrument. What's your favorite instrument in your lab or otherwise?
It's got to be either our electrophoresis instrument, so that's how we do some separations, or the mass spec. The mass spec the department has that we're using quite a bit now.
All right. The other question: do you use a Raspberry Pi in your lab? You do. You mentioned that.
We do. We sure do. So these are kind of we actually have a Raspberry Pi hooked up to a system, and that just kind of, we had an undergrad in our lab who was, she's now a graduate student in the computer sciences program. She kind of programmed the Pi to actuate one of our valves for us, and so it just runs continuously for us. We don't have to do anything. Look at that!
Getting hits. And a star.
Oh, he's gonna do this for like the last 2 minutes and just
block.
It doesn't. There, go, baby.
If you get a star off, I'll consider this nice. That's a victory. We're calling that a win. You're going the distance with Mike Tyson.
A lot easier with the cheat codes.
Oh, that's fun. Bronco made a mini arcade cabinet that runs an emulator on Raspberry Pi. Yeah, it does not take much to run these old retro games. LIMS system. We use a Pi for our LIM. What is that?
It's your laboratory information management system or something like that.
I see. So it's like a spreadsheet.
Collects all your data from your computer or from your instruments and kind of compiles them into like your electronic lab notebooks, right? Is that right, Oodles? Just a way to collect all the data and stuff.
All right, so what game is next?
What do you think? Castlevania, Super Mario Brothers, 1 or 3. Q*bert. Let's do a little Q*bert.
All right, Q*bert it is. I'm gonna mute you and let you change up. All right, so anyone joining us, we're doing a quick change between games. In the meantime, again, the guest today is Mike Roper. He's an analytical chemist and expert in mass spec, microfluidics, lab on a chip technology, studying cell signaling and diabetes. So if you have any questions, please throw them in chat, and we're happy to answer them. And Mike will be back right after he gets his game up and running. Oodles asks, Ken, how do you get your guests? So I used to run an in-person Ask a Scientist event, and I did that for about 6 years. So we had in Tallahassee, we have something called First Friday, which was basically when I first showed up, I went there and I saw, it's musician, artists, food truck, whatever type, once a month event. And I found out that anyone could set up a tent. And so we set up a tent and it said Ask a Scientist on it. Me and 4 or 5 other scientists just sat around and drank beer and talked science with people. And so over time, I had a couple news stories on it through word of mouth. I've slowly collected these experts across many different disciplines. And so I have a lot of chemists, but also we span astrophysics, biochemistry, a whole bunch of different disciplines. And then we moved it online. And so the guest pool of scientists that play video games is a little bit smaller, but still a lot of people excited to play. So there's a link in our in chat to our YouTube channel if you guys want to check out some of our old episodes. We have developmental psychologists, we have astrophysicists, whole bunch of different disciplines. Our previous guest was a sociologist who studies sea level rise with climate change. So yeah, a lot of diversity. And our next guest is going to be oceanographer Angela Knapp. So March 31st, Angela is going to join us. She studies nitrogen cycle in the ocean and marine life and how it's affected by nitrogen, as well as various different aspects of that. And so she's a, oceanographer / marine biologist type researcher. So that'll be March 31st. All right, Mike, do you have a game? No worries. Yeah, eventually the goal of this channel is to do once a week. Yeah, Mike's troubleshooting his controller and game right now. So one of the difficulties with remote changing between games, we're doing a Zoom link which shares screen and video. And so it's a non-trivial troubleshooting process sometimes. All right, Mike's still troubleshooting. In the meantime, any questions for me? I will pretend I know something about lab on a chip and microfluidics. Or more appropriately, color chemistry would be my best contribution. If not, we can do a random trivia prediction right now. Oh, totally not. Eugene wants to know, what's the most important instrument in your lab? Well, that is my computational chemist is the most important instrument in my lab. No, ours is the UV vis, just checking what color things absorb. That is our go-to favorite thing in the universe. What's your favorite color? I'm going to go with green, 532 nanometers. It is the second harmonic of a neodymium mag laser. It's a wavelength we work with constantly. Anytime you use a laser pointer, that green, that is 532 nanometer green. Very clean, beautiful green color. It's high intensity in the peak region where your receptors receive green. It's some of my favorite colorations. Ken, any work on fluorescent labels of proteins? No, my group has not done much in terms of biolabeling and things like that, but that's just because I don't know biochemistry and I haven't worked with many biochemists. I would love to. That's an area that, I wish I understood biochemistry a little bit better just to know where the gaps are and what problems need to be solved. Most of my work is in solar cell related things, so converting light to electricity, but also a little bit of catalysis driving reactions with light and things like that. But yeah, fluorescent labels are fun. We did publish one, it was with Tyler McQuaid in a collaboration where the molecule, when it came in contact with a primary amine, turned on emission. And so it was a fluorescent sensor for primary amines, and they actually threw it inside a cell and sensed amines. So yeah, pretty fun area. I'm excited to try to get into that. Yeah, I promise we tested this about 2 days ago and everything worked, but Mike's... Ooh, rural juror. Both Ken and Mike, how did you decide where to do your postdoc? Every one of my research decisions has been based on the type of research that they do. And so I worked for Mark Thompson as a grad student working on OLED technology and photo detectors. And as a postdoc, I wanted to get into the water splitting and photocatalysis domain. And so the big dog in that field in inorganic is Tom Meyer. And so that's what kind of drove me to Tom Meyer. With that said, I reached out to 10 people for postdocs and got opportunities from 3 of them. So part of the answer is you go where people are willing to take you. Yep. So the game's crapping out. All right. Yeah, everyone, I apologize for the technical difficulties. His USB controller is no longer working. So we're going to bring Mike back without the game, and maybe I will get a game playing. All right. So, Mike, you're back. We'll keep the audio silent on yours, but you added a few questions in the meantime while we troubleshoot a little bit.
Yeah.
Yeah, just go ahead and mute that game.
Sorry about that.
Yeah, Oodles recommends deleting System 32. Have you given that a try? He's working on a Mac. That doesn't work. Eugene says you should upgrade to Linux.
Thanks, Eugene.
That's fair. All right. The question was, how did you decide where to do your postdoc?
Yeah, so I did my PhD at the University of Florida. My last year, my advisor moved across the country. He moved to the University of Michigan. So my last year in grad school, I spent at the University of Michigan, although my degree is actually from UF. And so the people I was most interested in working for, I went and interviewed with them, and 1 or 2 of them they told me, they were very nice, and they told me they were about to leave their current institution. And so I was like, I do not want to do that. I don't want to make another move. So I chose another person. That being said, I was in my PhD when I was working with microfluidics there. I had started kind of doing microfluidics in my PhD lab. And I was the first one to use them in that lab. And so we were doing very clean samples. We kept clogging them. When we have really small channels, they're kind of a nightmare to work with. And so they would continuously clog. And this person that I went to work for, the guy who was doing these kind of interested in the rape kits, he was running blood through his devices. So he was just using really messy samples. And I was like, man, are you kidding me? How can you do that and not clog your devices? And so that really intrigued me, how he was using real samples and things like this. Plus he was stable. He was going to stay there for a couple of years, so I didn't have to worry about making another move. So kind of like what Ken was saying, the research drove me, but also there was some aspect of I don't really want to move again. Any sad stories on scientific recognition being largely misplaced scooped? Example like Rosalind Franklin and DNA structure. Whew, calm down, Bronco. Well, I have to say that I have never had, I've never had as bad of a problem as Rosalind Franklin did. That's for sure. Certainly, I think we've all, if you're in science long enough, it's hard to come up with a super original idea, and so people are working on the same topics as you all the time. So it's quite common to, not common, but it definitely happens where you will get scooped. There's no doubt about it. It's kind of like if you ride a bicycle, you're going to fall. If you're in science, you're going to get scooped. But nothing, I guess nothing has been to the point of like where we couldn't continue on with the project, right? We were always able to at least get some kind of a paper out of it.
All right, so Mike's computer is no longer working, so Mike, you're going to keep track of chat, and I'm going to play Mario while you talk science with people.
Cool. I'd love to do that. That's fine.
We'll have some background noise. It'll be fine.
So all right, so I can stop sharing my screen or just keep it up?
No, keep sharing.
Okay, that's fine. Delete System 3 and upgrade to Linux. So what about you, Ken? You have any sad stories on scientific recognition?
Oh,
Misplaced or scooped?
No, there's one particular one. You guys might have heard of the Thorpening effect. It's an organic chemistry. It's taught. It has to do with like orbital overlap and how reactions happen. Thorpe and Ingold were the PIs on the paper, and the first author on that was Beasley. And so Thorpe and Ingold went on to get Nobel Prizes. Beasley ended up dropping out of chemistry. And so 3 authors, 2 of the names got on there, despite Beasley doing most of the work, did not get recognition for their contribution to that. At least not as much recognition as Thorpe and Ingold did. So not too tragic, but still a bit frustrating if you're Beasley.
Yeah. It's pretty common, right?
I mean,
I feel like grad students do all the work, postdocs do all the work, and the faculty get a lot of the credit.
Yeah, oh, exactly. Like Tom Meyer he was like, "Oh, you're here to make me famous." I'm like, that's fair. Like, that's what we do as grad students and postdocs.
So how long, Calm Down Bronco asks, how long does a postdoc typically take?
Oh, man.
So it varies depending on the field. There's biologists who spend 5 to 6 years in a postdoc. That's fairly common for their field. They can take that long, especially if they're, let's say, they have to make some genetically altered animal. It takes a while to get those and have them be able to breed and be able to get the fruits of their labor, essentially, before they can do any experiments. Chemistry, I think, I'm guessing 2 years is the average, I would guess. Is that the same for you, Ken?
I think it's getting longer. I think 3 years is the standard. I mean, at least it depends on, if you want to go into an academic job, it's very different than industry. Like industry, you don't even necessarily need a postdoc, but academia, you have to build such a foundation to even be competitive for a job. It's just, yeah, you have to spend 3 years just to get those papers out so you can actually be competitive on the job market.
I do find that the faculty candidates we interview have much better resumes than I did when I came out as a faculty candidate. Right? They have a lot more papers, maybe bigger journals. Maybe it's because there's more journals now than there was 15, 20 years ago. I don't know. But yeah, so a third year postdoc would certainly help for sure. I actually spent 3 years in my postdoc. I think it definitely helped.
I mean, and postdoc is kind of an awesome research experience because you have none of the responsibilities of like finding money and maintaining a group and running it, worrying about whether your grad students are successful. And you don't have classes, you don't have hoops to jump through. Like, postdoc is pretty awesome. Or at least it was for me. I don't know about you, Mike.
I worked with some great people, and I met my wife there, so I have to say my postdoc was great.
That's fair.
But I worked with some great people, and some of them are like faculty now, so I think we had a great group of people. I do remember going to my postdoc advisor and telling him that I did not want to be the group psychologist because of like the group of students I was mentoring. And he just, I remember him just laughing and saying, "What do you think a professor actually is most of the time?" And that's kind of stuck with me also, and I think it's pretty true, right? Like you have to figure out what motivates people, how to get them to do some of the experiments that you want them to do, or to kind of think along the same line. So I think it taught me a lot of how to mentor students, for sure.
Yeah,
good question here from Oodles of Omics. It's a good one. Favorite conference location? It's got a double question, but I'll start there. I have a favorite conference location. There's a small conference in northern Italy, in Riva del Garda, Italy, which is right on this lake, Lake Garda. And it's really a difficult conference to get to. It's a difficult location to get to, but it is completely worth it. It's beautiful. It's kind of nestled in these mountains, kind of the foothills of the Alps, essentially, and it's just so picturesque. It's beautiful, and Italian food is just delicious. Like real Italian food is delicious, and people are super nice. And it's in May, so the weather is perfect, and it's just great.
Yeah, I had a conference in Rome. That was pretty good.
Yeah.
That'll do it.
I like it, man. Went to a conference in Dublin a few years ago, and that was also a lot of fun, too.
So the last conference I went to was actually in the Bahamas last February. But it was like an all-inclusive resort that was not particularly great. So it was a super anticlimactic trip to the Bahamas.
Is it like a Sandals?
Kind of. I don't remember the name of it, but it was not great. With that said, like, what an awesome job we have that we get to actually travel and do and talk to our friends.
It is, that's true.
I mean, it's such a luxury that at some point we get frustrated that we have to do it, but it's pretty awesome. I'm
pretty lucky.
wrecked.
And Roro Juro just called out my conference. Yeah, that's right. ISCC. That's the one I was talking about. GCGC is also there. Right. The other question Oodles of Omics asked at the same time, that same question thread there. If you could change one aspect of the scientific field as a whole, what would it be? I know exactly what I would say. I hate the competition aspect of it. I feel like there's a lot of people would much rather be first or be the one to do it rather than see the science get done, maybe properly or collaboratively. And so you get a lot of big egos, I think, in science. And it's not always best for science, if you ask me. So I don't know how to change that, but that would just be a pipe dream. Gosh dang, Ken, you're pretty good.
Yeah. Mario, I have Game Genie on. I have infinite lives, but I'm doing a bunch of speedrun strats here.
Yeah. Eugene said you didn't wait for the bullet.
Yeah, there's a bullet glitch through the flag. So I don't know if you know this, Mike, but Eugene and I started speedrunning Mario like 2 years ago. So we had a competition who can beat Mario the fastest.
And?
I'm winning at 5 minutes 2 seconds, 0.73. And Eugene is at 5:13, something like that. So yeah, I was ranked 176th in the world, and now I'm like 280th or something. That many people are still playing this game.
That's crazy. I did know that because when my students told me about it.
Yeah.
I told them that we were having this thing tonight, and I was like, do you guys know Ken Hansen? We're supposed to play games together, and somebody was like right away like, did you know he was ranked like 178th in the world and like Speed Mario?
Yeah, I played a little bit of and I'm number 9th in Tecmo Bowl, and I will be number 49th in Dr. Mario when I submit my actual video. So yeah, I play a lot of retro games. But anyway, for anyone just joining us, Dr. Mike Roper is the guest while I play Mario. He's happy to answer questions.
This is me playing. This is me playing. I'll just put on my...
And I'm just staring randomly off into space. Oh, I'm ranked 273. Oh, come on, Ken. Hold the button. There's a 2-frame jump here if you want to go fast.
Hey, you didn't answer the question, Ken. What if you could change one aspect of the scientific field as a whole, what would it be?
It's the money issue. Like, we just scrounge for money. Like, make no mistake, if I won the lottery, I would still be back in work doing exactly what I'm doing. I would just have more money to do it. You know, it's like we do this for a passion. We don't do this to get rich or anything like that. There's other ways to do that. But we have to fight for every single penny and dime and, you know, grant we get. We submit 10 and hope one of them gets funded. Like, it takes a lot of time and effort away from research to get money to do research. And so that's probably the most frustrating part about what we do. At least to what I do. No, fireball.
Well, we have faculty at Florida State, right? Bob Holton, who essentially did win the lottery, right?
Yeah.
And he did go right back to work, you know? He never retired or anything.
Yeah, so Bob Holton, he came up with the synthesis of Taxol, which is an anti-breast and ovarian cancer drug. And what did that patent make? It's something like 580 million over 15 years. And he got a good chunk of that towards his research. And he just like stopped applying for grants and just used that money for research. Like, it's amazing.
He got to write a 2-word letter to NIH.
I did not hear that story. Well,
no, I just he never had to apply to like funding ever again, right? He could fund his own research.
Yeah, that would be the best case scenario.
There was a story I heard from a colleague who, when Bob Holton, I guess, got one of his first royalty checks, he came to him and he said, you know, I got my first royalty check and I went to the ATM to deposit the check. It was after hours.
An ATM.
And he said, do you know that the maximum amount you can put in for a deposit is 99,999 and 99 cents?
I did not know that. Thank you for asking.
The guy's like, no, I did not know that, right? And so he said, like, the next morning at like 8 a.m., the bank president called and said, Dr. Holton, you don't ever have to worry about coming to the bank again. We will come to you and get that check for you.
That's amazing. That's FU money right there.
That's what I'm saying. Yep, for sure. Yeah, I agree. The money chasing is hard, and I'm on a grant panel, and we see a lot of really good grants come in, and you know, very few of them get funded. It's a tough game. Let's see.
There we go.
Ninja Physics 44 says, speaking of conferences, I just saw that APS just declared they wouldn't meet in cities that have bad police records.
Wow.
Anything like that in the biofield? Boy, not that I've heard of. It's really interesting. That's fantastic. I bet you more and more conferences will start doing that, though. Right. You did it, Ken. Nice work.
I'm going to do one more Mario run and we'll decide from there. Yeah, I jumped through the hammers. So Nintendo can only process something like 5 or 6 objects on the screen at a given time. And so if there's 8 hammers on the screen, like 2 of them don't really exist, so you can jump through them. So another fun factoid. Right? Yeah.
That's cool. I didn't know that.
Yeah. Oh, 6 fireworks.
There's a question from Oodles of Omics. Ken, have you ever played Kaizo Mario?
I have played some Kaizo Mario. So I actually play a lot of Super Mario Maker 2. And so I love the shell jumps and double shell jumps and shell drops and all that stuff. I played it on stream when I was going, but I had a really jank level during that experience. But yeah, I love Mario Maker 2. That's my go-to modern game right now.
That's
So those of you not familiar,
what is Kaizo? What is Kaizo, Mario?
So Kaizo, basically they figured out, this is probably what, 15 years ago, they figured out you could look at the code of Mario and write your own Mario games. I missed the jump. And they basically made Mario super challenging. Like you can play high level, expert, crazy levels like this. These levels are easy once you get used to them, but there's all sorts of tricks and stuff you can do, and they figured out how to write games that would really tap into those hard levels. And then Nintendo released Mario Maker, which allowed anyone to make their levels and upload them to a server, and anyone could play them. And so, yeah, that's...
Do you
Mario
make your
Maker.
own levels then, or you play other people's?
You can do either. I only play other people's levels. I'm not going to claim to be good at designing levels. But, yeah, I really love the... Some people come up with some creative stuff. Others come up with absolute garbage that you have to play anyway. So, it's a give and take.
Yeah, Oodles of Omics made a good analogy. Basically, if HF were a Mario game.
That's fair.
That's fair. All right, so then Calm Down Bronco asked, do you have any general advice for an aspiring Chem grad student? And Eugene responded, work hard, play hard.
That's good advice.
That is good advice.
No, the number one thing I'd say is get in a lab and start doing research. One, to figure out if you like it, and two, to figure out, or to get your resume built up, to have research experience and whatnot. Oof. Sorry, Mike, we've reversed roles. What's your advice?
No, I would, I'd agree with that. Do research if you're... I don't think you have to keep an open mind too, I think, especially in science. So if you, like when I was doing undergrad research, I found that I did undergrad research in analytical chemistry and electrochemistry, but I knew that the type of research I was doing as an undergrad, I didn't want to do for the rest of my life. So, you know, let that be okay as an undergrad. Let it be okay that you can switch fields, you can do whatever. Just find what is going to make you happy and make you passionate, so that you don't find lab work to be a struggle or a pain, you know, as you go into grad school. If you enjoy it, then it's not a big deal to come in, you know, work the long hours or to read papers or what have you. And then you can play hard, right? I was doing pretty hardcore electrochemistry as an undergrad. And I really liked electrochemistry. I love the idea of like, oh, if we apply a high enough voltage, we can oxidize or reduce anything. And it was just, I really loved that idea, but at the same time, I was like, man, this is getting a little too physical chemistry for me. No offense, Eugene. But the research that I was doing. So, and Hamsterlicious88, it's a good username. I went from biology to chemistry to mathematics and then to physics before I finally settled. Yeah, totally.
It's interesting you went to math first and then physics. I would expect it the other way around, but like getting narrower and more focused on fundamentals.
Yeah. Ninja Physics says, I did my master's thesis on hurricanes, which no one was doing research on in my department just because I wanted to.
That's fun.
Probably a good thing to do research on. Here in Florida, at least, right? Just watched Twister the other day.
Documentary filmed in real time.
Seriously.
Oh man, I should start busting out some of my questions for you, Mike. So
I got some.
So
Go ahead.
The one I ask every time, and this is one I want to know, and it's for me. What movie or TV show gets your discipline right, and what gets it wrong?
Ooh, definitely CSI. There's a lot of people who got interested in like forensics, kind of the analytical chemistry side of forensics. And they got some things right and some things wrong.
Well, you mean you don't just put something in the mass spec and it shows up on your screen?
Right, exactly, yeah, or tells you exactly,
structure.
shows you the, like the molecule in like 3 dimensions, like exactly what you, what it was. Even, you know, did you ever watch Making a Murderer?
I'm familiar. I didn't actually watch it.
You didn't watch it? So that's a Netflix thing where they Netflix docuseries that they followed this guy who supposedly was whatever. He's in jail right now for committing a murder, and so they showed kind of the documentary of like his trial and all this stuff.
Mm-hmm.
And so I couldn't help but laugh at that one. Now a lot of the stuff like you see in the trial, some of these lawyers presenting like some of the controls that they didn't run and things like this, you're just like, boy, as an analytical chemist, I certainly would have asked for that control, right? So yeah, I don't know too many TV series that get us right too well, but there's a lot that get us wrong, I guess. What about you? Do you have any? Like I don't watch a lot of TV, so.
Well, Breaking Bad is the one that gets it right.
Oh, Breaking Bad is a great example. Yeah, for sure.
Yeah, that gets synthetic chemistry right. A whole lot of aspects of it. Oof, it's pretty gross.
Well, they had a great scientific advisor on that one too, right?
Yeah, they had several. One of my lab advisors from the University of Southern California was on their, I don't know, advisory board or whatever they want to call it. But yeah, they did their homework. They... I mean, basically any movie that shows molecules on a chalkboard at some point has carbon with 5 bonds and, you know, hydrogen's double bonded. Like it happens everywhere. I thought about like incorporating that into my Gen Chem class, like what is wrong with this and how do we fix it kind of thing. But
I talked about the battery that they make on Breaking Bad.
Yeah. Which class do you talk about that?
In electrochemistry, in the analytical chemistry class. So we do electrochemistry, so we like show galvanic cells, and you can calculate the voltage, and it's pretty close to what you'd need to.
That's awesome.
I don't know the current that would be required to start the car, you know, but the voltage is about right.
That's awesome.
All right, hang on. Trying to catch back up to some of these questions. So there's a lot of discussion about math. Pure math, oh gosh. Applied math. What is the most difficult aspect of mentoring graduate students? How much time do we have?
We have 1 hour and 13 minutes.
There's a lot of good things about mentoring grad students, don't get me wrong. So, but the most difficult, I guess expecting them to be like you were in grad school. Having wrong expectations. We all have different motivations, right? So assuming that they have the same motivations as you did when you were in grad school, and learning that everybody is different and having different expectations. So I'm just trying to, you know, normalize your expectations, using a word from earlier.
That's in teaching too. Like when we write a test, it's very hard not to look at a question and say this one should be easy. It should be 80%. It comes back 60, and it's very easy to lose perception of where other people are and what their experiences are, and people are individuals. It's definitely a difficult part.
What about in COVID time? Have you had any? What do you feel about mentoring grad students in this time, Ken? Are you going into the office?
No, I've been entirely at home. entirely at home, but we have group meeting once a week plus one-on-ones whenever they request it. But yeah, it definitely feels much more detached. It's kind of sad, but
Yeah.
I don't know. They seem to be doing okay, but I think all students are kind of just losing it after a year of this.
Yeah.
We're coming up on the 1-year anniversary of our first shutdown, right?
Yeah. Daily motivation battle for sure.
It's very real.
That's what Hamsterlicious says. Have a daily motivation battle. I think that's absolutely true. I think it's. I do the same thing. I do like Zoom weekly meetings. So that we have group meetings, and then I try to have a individual meeting with each of my students every week, just to kind of check to see how they're doing, what their data is, and stuff like that. But it's tough. Like it's kind of weird. My students will be like, "Mike, are you going to come in?" I'm like, "Yeah, I'm going to come in. Thursday. I'll come in." And I think they just want to make sure I'm actually just not this two-dimensional figure on a computer screen.
All right, Tecmo Bowl it is. Mike, I'm really sorry you didn't get an opportunity to play these games while talking science, but I appreciate
I'm
you
really
sticking
sorry
around to
that
hang out.
my controller didn't. I don't know what happened.
Yeah, it's crazy. We tested all this yesterday or 2 days ago plus today, and yeah, so it goes. So I'm going to try to show you a Tecmo Bowl speed run. So when you speed run Tecmo Bowl, the goal is not to win by as much as possible, but to kill the clock as much as you can. So you basically try to stay from getting tackled as long as possible. It's a very different game type.
Okay.
So don't look for high scores here. I'm going to win by 3 is what I'm going to try to do at least.
So is this Chicago with like Walter Payton?
Yep.
Nice.
So you just try to kill as much time as you can, which means the goal is to not get tackled. Narc speedrun, yes, Bronco, it's coming.
All right, so we got another question here that maybe I can't answer very well, but I bet you can. Maybe. I keep hearing that we are nearing the max capabilities of current battery technology. Is that true?
Whew. Like lithium ion versus, I think we are pushing lithium ion to their limits. So people are like, there's oxygen batteries and there's all sorts of different ones. I don't know the nuance of that. I need to have Yan Yan on. I know she plays video games, but she would be the go-to expert. She's working on transport mediums for batteries.
Running the wrong way. Oh my gosh.
Oh man. No, I'm going for it. Sorry, we're trying to do chat. Oh, they picked it. I'm in trouble. So this is a risk-reward game, and I just lost. That's fine. We'll play defense.
Oh, there's a question, a channel question, Ninja Physics. What is the lead time you have for notifying subscribers that you're streaming?
I think the notification only comes out when I send out when I start streaming, so it's the 15-minute warning. But if you follow our Twitter account, I give 1 week before the guest, and then I'll give a day of warning who the guest is. So yeah, follow us on Twitter. That's where we do our notifications at least a week ahead of time. Also, if you look down the page on schedule, you can see our next one. It's going to be Angela Knapp. If you just look at that, that'll tell you who the next guest is and when it's going to happen. But yeah, eventually sorry, go ahead.
No, go ahead.
Eventually, when we can do this in person, I want to do it once a week on a set day. Because that would just be easier for everyone. Because then I could just run it out of my house and have people come over once a week.
So these aren't set days? I had assumed they were.
No. It's. I mean, we're so flexible right now that.
Yeah.
This is Tecmo Bowl at its finest.
You know what I hate is that I'm a big Cowboys fan, so I hate to see you doing this to the Cowboys.
So it turns out Chicago versus Dallas is the best game, and it's because Dallas's offensive line, watch them just get shed by these blockers.
Dallas was that bad, huh?
Yeah.
Hey, so why don't you, that's what I would love. So if we do this eventually at your house, we can play each other.
Yes. Feasibly we could, yeah. You want to play Tecmo Bowl? Let's do it. I'm excited.
I thought for sure. See, that's what I thought. Bring out Bo Jackson also. See, when you first invited me to do this, I was telling my kids, I was like, all right, I'm going to be playing Ken, but he's like really good. I don't think I'll be able to beat him very much.
I mean, it's something I wrestle with because you want to keep track of chat, and it's not trivial to do.
Yeah, it would be really difficult, wouldn't it?
Yeah.
I don't know if Eugene is still here, but somebody, Hamsterlicious88, is still saying that they're trying to learn quantum field theory right now. It's painful. Of course. Why would you try to learn that?
Man, I don't even know where you start with that. What's a good...
Eugene has left us, I bet you. So if you're just joining us, I am not Ken Hansen.
Sorry, I'm distracted.
I was supposed to be playing, but my Mac decided to.
I mean, you could bust out
the USB controllers.
You could do Qbert with the keyboard. I mean, that's only directions, right?
Yeah. I'll do whatever. I mean, I'm happy to sit here and talk and drink, and it's fine.
Yeah. Watch speedruns in Tecmo Bowl.
Oh, calm down, Bronco. Asked a question. What experiment would you run if you had unlimited funds and morals don't matter? Whew, let's see. Gosh. It's a great question. What is yours, Ken? What do you have?
Oh man. I mean, I'd love to do some kind of massive like Manhattan Project with solar cell related research. Like just get everyone in one space and give $1 trillion to, you know, just try to fix it kind of thing.
Right.
But that's the morals one. I don't know. That is an interesting question.
Yeah, so I'm very interested to understand how cells talk to each other. So the cells in the pancreas that we're really interested in, there's actually a collection, these islets that I've talked about. They're collections of cells, and they're spatially connected to each other in certain ways, and we don't exactly know why they are the way they are. So I'm always trying to, kind of a goal of our lab is to understand how these cells are communicating to one another. So if we could design a way to try to look at all the different things that they're releasing to within the different cells or to each other, and to me that's like a holy grail, to try to understand proteins, peptides, small molecules that they could be releasing to communicate to one another. I think it would be super fascinating to try to listen in on how they're communicating to each other. Again, I don't really have a morals.
Just say you'll do it on stem cells. then it's okay.
Yeah.
So unlimited budget, what do you buy to make that happen?
Gosh, good question. A lot of people. You just need a lot of fans.
An army. Yeah.
People are probably the most expensive thing that we
yeah.
that we need. I think a lot of the technologies, and yeah, so somebody asked a question, Oodles of Omics asked if we've worked with lateral flow assays or ELISAs. Yeah, we've done some ELISAs. We try to stay away from that. We don't like ELISAs so much because they're slow and pretty laborious. So we develop our own type of assays to try to kind of bypass the downsides of ELISAs and try to have our own immunoassays going on in our microfluidic systems. I think, yeah, if we'd have to like make really small instruments to kind of make sure dilution isn't too big of a problem of those biomolecules, so you could still measure them essentially. Maybe possible one.
Speaking of dilutions and concentration issues, so the COVID test right now, the most accurate one is the PCR experiment, right? And so PCR, they're just amplifying the material so they can measure it. Is that right?
Yeah.
So, I mean, what are the limits of what could be measured? Can you do it without PCR? Is there some instrument that could do that? It's just not time and cost effective?
What I know about the levels of the virus in the blood, it's so low that you can't measure it without an amplification mechanism. Mm-hmm. It's extremely low.
So like your best mass spec couldn't see it, or you couldn't?
No, and mass spec certainly doesn't have the best limits of detection. Like probably optical detection or electrochemical would still be even better limits of detection. Even those don't, unless they have some kind of an amplification mechanism, it'd probably be too low to detect there. And that's the great thing about PCR is it amplifies, right? And you're able to detect it. And I think that's a big reason these point of care antigen tests aren't very. I think if you look at the specificity, at least the one that I've taken, those rapid antigen tests, I think the sensitivity was 56%. So, right, sensitivity is essentially like how correct of an answer it's going to be, right? Like so 56% is not great, but we got the results driving home from the testing site, so it's super fast. Yeah. And I think that's why that it's so low. It's 50. We'd much rather have like a 99%, 95% sensitivity. But that's why you can't beat the PCR right now. At least, especially the PCR test in that Florida State is running, they amplify three different pieces, strands of the segments of the virus genome there. So I think they call a positive if you have all three amplified, and it's inconclusive if you have just one. So when they call positive, they're extremely positive that it's you've got the virus, right?
And if you have a negative, you still might be positive. It's just not certain.
Well, I think the negative, you have to be negative in all three of those strands. So if you have one of the strands that show up positive, they say it's inconclusive, so they ask you to come back. So they don't call you negative there. So I think with the negative, you're okay.
I just got scored on. What is this madness? Sorry.
You need a drink. Somebody needs to buy. How do I buy a Ken a drink?
Your internet points? Click on the Xi symbol.
How would I know that?
It's right under your chat. Go down to your Xi. Oh god, Oodles and Bronco just redeemed. I actually have to go get a beer, so I'm going to pause. And Scuzzbot. All right, we're hammering an entire beer when I get back.
Oh, there you go.
I can't pause in Tecmo Bowl. I need to make that a higher priced item.
Now I'm doing them. Yeah.
There we go.
You got like 4 times. Yeah. Hamsterlicious, you asked, do we simulate the microfluidics? We do. We use some finite element analysis to simulate some of the flows in our devices. ComSol. Again, because the fluid physics at these small dimensions are pretty well behaved, it's fairly simple to simulate those. So a lot of times we'll think about like what kind of reactions we want to do or what kind of assay we want to do. If we want to do an immunoassay, we know we want to do certain things with our reactions. So we'll have different channels come together and we can simulate that before we go and spend time fabricating the device. Boy.
All right, Mike.
Ken, you are.
What?
I hope you have a couple beers there. What are you drinking there? Are you still drinking the
No, Blue Moon.
proof?
Blue Moon.
Oh, Blue Moon.
All right. Well, here's cheers.
Cheers.
Mike, you're drinking too?
I am. Wine. I switched over to red wine.
Keeping it classy in here.
Trying to.
In person,
Gotta bring some class.
I need to do like a 10,000 internet point shot. We'll do vodka. But anyway,
Cheers.
while I pause the game briefly, everyone, thank you for stopping by. We appreciate talking science and playing video games. It's, a lot of fun. Mike, it's great to have you here. Our expert today is Mike Roper, expert in lab on a chip, bioanalytics, diabetes, microfluidics. Any questions you guys have related to analytical chemistry, he is the go-to guy on answering them. So feel free to throw your questions in chat while I finish this game of Tecmo Bowl. So this is third large drink. Hamsterlicious, thank you for the gifting 5 tier 1 subs to the community. We really appreciate it. So yeah, Ninja Physics, Faithful Fury, Fluidics, Guzzbot, and A-Rob. So there's follows. Yeah, the subscription hype here. So Mike, if you're not familiar with Twitch, we have follows, which is people that follow the channel and get notifications when we go live. People can also subscribe, where they essentially donate money to the channel to help maintain it and, buy beers for people. And so yeah, Hamsterlicious just donated 5 subscriptions to members of the community. So we really appreciate it, Hamsterlicious.
Awesome. Thank you.
Yeah, clearly he or she was super impressed by your 48 seconds versus Mike Tyson. Worth the subscription, worth every penny. Oh man, I'm sorry we didn't get to go to Castlevania and Qbert.
Did you ever play Castlevania?
What's that?
Had you ever played Castlevania?
I have. I just there were certain parts of the game that required knowing the pattern, and I never looked it up or learned it.
Yeah.
Where you have to go. Do you want to give Qbert a try on keyboard?
Okay, finish your game and then we'll do it. It's fine.
All right. Oh man, I didn't even realize I'm running out of time here. All right, game over. Loss.
Did you lose?
I did. I was just trying to kill time. So there's a category for speedrunning a loss as well. So this was probably, 15 minutes with the pause? But, all right, you're going to do it?
Sure.
All right, so I will transfer to this. Go ahead and share your screen and figure out what your keys are going to do.
Hang on, I'm going to stop share, Ken, because I've got to switch over to a different.
Yeah, that's fine.
Okay.
Mike, I'm going to mute you momentarily while you troubleshoot that. But again, everyone, thanks for joining us. If you have questions for Mike Roper on analytical chemistry, throw them in chat. Yeah, it's great to have everyone here playing games or talking with us while we get to play games and drink alcohol. Flashbacks to undergrad and graduate school. So yeah. Way less formal seminar. I agree with you, Hamsterlicious. This is the antithesis of the formality of seminar. But also that's one of the reasons we like to do this, is that there's a certain ivory tower aspect of academia where it's like you have knowledge and you seem impersonal in this fountain of knowledge that you have, but really most of us are just people that like to drink and play video games and shoot the shit, and this is a mechanism that we can do that with the general public. So yeah, we're really happy to do Ask a Scientist Gaming. People are super enthusiastic to join and share their science with people. And yeah, when we don't have to do it via Zoom, this is going to be way easier, but right now we'll do it remotely. Yeah, I think when we're vaccinated, I would feel much more comfortable having people come over to do it. Because I think Fauci even said if you have two people that are vaccinated, they can hang out and there's zero concern. Not zero, but negligible concern. Ninja Physics. I'm supporting with my temporary non-academic salary. Yeah. If you want to make money, do not go into academia. It was mind-blowing for me when I, worked really hard to get this academic job, and then my first grad student made like 30% more money than I did right off the bat. I was like, you deserve it. Well done. My advisor told me I don't do grants and I'm bad at networking. Harsh. Yeah, networking matters. Having friends matters. It's pretty brutal. All right, Mike is troubleshooting Qbert and how to do the controls via keyboard because the controller isn't working. No, we don't have a Discord for this channel, but yes, there should be. I got to figure out how to do that. If somebody wants to volunteer to set up a Discord for this channel, send me an email, and yes, we should definitely have one. It's just, yeah, side project, but the remote streaming we've been troubleshooting a lot right now because it's a non-trivial process. You're on? All right. Screen share, sound, and video. And I will make sure to get you back on screen. Oh, man. Volume's good. Turn your volume up a little bit. All right, we're back with Professor Mike Roper playing Qbert on keyboard. It's just 3 arrows or 4 arrows, right? So I don't hear any sound, Mike. There we go. Perfect. All right, there we go. How does this game work, Mike? That is the most important question you've been asked at this Ask a Scientist.
How does the game work? You're supposed to... Well, not like that. You're supposed to...
Infinite
You're supposed
lives?
to jump on every square. Son of a... And change its color. See, I'm trying to change from blue to yellow. And it's more than a little difficult to remember. Like, right arrow actually goes down and to the right. So then left arrow goes up and to the left. So once you change all the colors, then you move on to the next level, and there's like more people that come out. Dang it.
My favorite part of the game is when Qbert swears when he gets killed.
Hey, can you give me just a couple minutes? I say goodnight to my kids, and then I'll be
all
right
right.
back.
Sounds good.
Thanks.
All right. So I'm going to transition back. I should have changed the game. Anyway, does anyone have any questions in chat for, I guess, me, unless Mike's coming back?
I'm back.
All right. Mike is back. All right. So Mike's playing Qbert. Do you have Game Genie codes in? Infinite lives?
I don't think so. It's all right.
Is Qbert a game that's endless? Like you can't beat it, it just continues level after level?
That could be, actually. Look at this, and now it's...
reset. We got electrical engineers. We have chemists. Fairly sure we have an analytical chemist in lab. A Department of Defense lab working on multi-omics. That's really fun.
That is fun.
And we have a CPA in the house. Does anyone need their taxes
Sweet.
done?
I need to do my taxes.
Analytical
Hit me
chemist
up
working
after
on
this.
separations.
Who is?
Rural juror. Mike, did you know we published a separations paper?
Get out of here.
Yeah. No, it's photochemical separations of iron, ruthenium, and osmium. So we basically have
Nice.
it's a ligand you attach to a metal, and depending on what excitation wavelength, you get a metal to ligand charge transfer that transforms the ligand to something that you can separate.
I have so much more respect for you now, Ken.
I'll send you the paper because I'm like, I've published in Materials, Physical, Inorganic, Organic, but not an analytical journal and not a biochem journal.
That means you can teach our analytical classes now.
I wouldn't even be opposed to that. I learned way more about chemistry by teaching it than actually researching it. Like when I had to teach Gen Chem, like I really had to learn general chemistry. Like for the first time, really, on most of the aspects of it.
I completely agree. I think I try to teach pH, and I'm like, man, you guys just need to try to learn acid bases as if you had to teach it to somebody else.
Yeah.
It's the only way to really learn this stuff. Right? If you can teach it to somebody else, that means you've learned it really well.
Yeah, that's what I tell my students. Like, I feel bad when they ask me, like, what should I do to study for your class? Because there's no silver bullet. But my default answer is, like, call up your parents and explain this to them in an accurate way. And then you'll feel like you really understand it.
That's a great question. That's a great way to explain it.
Ninja Physics is an astrophysicist by training, but working on getting into public policy, clean energy policy. That's awesome. Bronco wants to know, how expensive is it to own/run a lab?
You know, I've done that calculation where I try to figure out how much each student spent. So biology is pretty expensive. So we do a lot of biology, and we do everything from, like, you know, animal surgeries to, you know, lasers and all that stuff. So we spend a lot of money. I think ours was like roughly $500 per student per month.
That's in materials.
Yeah. That's right. It doesn't include personnel costs.
Yeah. No, that makes sense. I think I'm somewhere around that range. I mean, what's crazy
though? Crazy range, right?
Yeah, I mean that's when you're operational. But like for us to do a femtosecond measurement, so ultra fast measurement, it's roughly a $300,000 laser just to get started with one measurement. And so it's really the big equipment that gets really expensive very quickly. Oh, that was kind of a bullshit death. What happened there?
That was a bullshit. Thank you for saying that. That was bullshit too. I couldn't remember what era it was.
Oodles of Omics wants to know, do you have any side hustles outside of academia? Like meth or, Mike, what do you do for fun?
For fun or another job?
Side hustles.
I'm an editor for a journal, so I get a little compensation for that.
I did not know that. Which journal?
It's an RSC journal called Analytical Methods. So I'm an associate editor for them.
I'm going to just rant a little
about
bit.
300 manuscripts a year.
I'm just going to rant a little bit. Like, in our department, we don't do enough to celebrate our own colleagues. Like, becoming an editor of a journal it's a lot of work, but it's also like an honor. Like, you've been recognized as someone in the community whose opinion matters. And so it's awesome when somebody gets an editorship. And I didn't know that, Mike, so congratulations, but also sorry.
Yeah. I've been doing it for about 4 or 5 years.
Oh, wow.
So get a little compensation for that, and then I review for NIH. I'm a chair of a study section for NIH, so I get but everybody who serves on the NIH panel gets a little compensation, but it's not really, you're not getting paid for that, you're more getting like just reimbursed. It's a totally volunteer sort of thing. But that's always cool because you get both of those are cool because you get to hear about some of the coolest science that's being done. Right? So that's always, it's almost like you have to keep up with the literature that way because you're reading about it constantly.
Yeah, so I have the same, like, sometimes you get paid to review stuff, but my favorite is serving as an external member of a thesis or a committee. So, like, Australia and most of Europe, they have an external member to the committee who's actually external, right? It's an expert from the field that's not part of the university. And so I've done that in Australia a couple times. I have one coming up in April in Sweden where I'm they call you the opponent. So they actually, if you're not
that's a big deal.
Yeah, if you're not COVID related, they'll fly you out. You come out there, you say, here's where the field is now, and then you're the one that questions the person for like an hour. So you are the opponent to the defense. And so you get a pretty nice paycheck with that, plus a flight if we're not COVID times. So that's kind of cool.
That's great, Ken. Congrats on that too.
Yeah, that was really fun. Timing's terrible, but I'll take it.
Yeah, because I think they treat you like really well if you're there, right? They like take you out.
Yeah, that's
like a good thing about academics. You get a little wined and dined occasionally, right? It's always nice.
Perks of academia. Yeah, no, it's true. It's such a luxurious position. Like, we complain about we're not going to get paid as much as industry, but the flexibility and schedule combined with the additional perks on the side, academia is an awesome job.
That's right.
Especially if you're okay or like teaching, it's great. All right, Rural Juror has a editor question for you. If somebody submits a manuscript with suggested reviewers, do you actually send it to any of the suggested reviewers? Keep in mind this video will be on YouTube.
It depends. It depends. Like if I know those people that you've suggested and I think they're actually competent reviewers, then sure, I don't have a problem with that. I usually won't send it to 2 of the recommended reviewers. I'll send to 1 of them. Just because I don't know everything about the author and that reviewer's relationship. So I'll try to just pick at least 1 other reviewer, if not 2 other reviewers that are my own choice.
I'll give you a hint. Do not suggest a reviewer that has a Gmail account. Probably won't go out to them.
No, we're stopping. Yeah, we're trying to stop that, actually.
Yeah.
But I've heard NSF, and I've just heard this as a rumor. I don't know if it's true. We'll definitely not send your reviews. If you suggest reviewers, they'll only send to 1 of those, and they'll essentially the other remaining people are then precluded from reviewing. So if you suggest a lot of people, you're essentially taking out a lot of their dang it, green guy. You're taking out a lot of your field.
Wow, that's interesting.
So yeah, be careful.
So Hamsterlicious didn't know you could suggest reviewers. So those of you not familiar, peer review process is essentially we write a paper and we're going to submit it to a journal. And part of that submission process is, the title, the abstract, here's the article. But they also ask for, 3 to 5 suggested reviewers. So it's people in your community that you think would be good at reviewing the text. And then they also typically have some kind of comment saying why you think this person would be a good reviewer. And then that goes to an editor like Mike, Mike, and he decides, is this appropriate for this journal? Should we send it out for review to our community, or should it get rejected outright? If they decide to send it out, they pick 2 or 3 people, or I guess 3 to 5, depending on the journal, and then they try to get reviews back from the community at large. And so it sounds like Mike will use at least one of those suggested reviewers typically, but maybe not. Maybe it's people you don't suggest.
Not always.
Yeah. And it's one of those things, this is something I wrestle with when I submit. Like, do I submit friends, or do I submit, the best person to review that I might know is kind of an asshole, but is going to review it in a critical way? Like, it's a cost benefit.
The problem is, like, we're all so busy, right? Mm-hmm. So I have a pretty, I'm the associate editor of a pretty small journal. So if I send it to like a big wig, I just feel like the chance that they're gonna say yes is pretty small.
Yeah.
So you saying that right there, like, you're gonna probably put a big name because you want them to see your paper. I get it, and I'm like, they're never gonna say yes.
They're never gonna say yes.
and then they won't say no either. And so what will happen is the email will sit there in that person's email box for 5 days before finally it's like, okay, this guy's not gonna, this lady is not gonna say yes. So finally it comes back to me after 5 days saying, hey, you need to choose a reviewer. And it's like, oh shoot. So already that manuscript I sat there for 5 days with no reviewers. So I just, oftentimes if it's too big of a name, I won't even ask them. But I'm not an editor for science or something like that.
Oh man, Ninja Physics has redeemed request a factoid. What's your factoid of knowledge you want to dump on the community?
Okay, hang on.
He has notes. He has notes.
2 letters do not appear in the periodic table. Those are.
Who wants to throw their guesses in chat? What 2 letters don't appear in the
periodic
table?
I was going to say, I hate to just give them up like that.
No. Let's see it in chat. Anyone have guesses? 2 letters. It's not H or B or C. What 2 letters don't show up in the periodic table? J, we have a guess for
dang
Q.
It, you guys, very good.
Is that it?
J and Q.
J and Q, congratulations, Bronco and Ninja Physics.
Acrylic alphabet. There's the
Hamsterlicious, it's lanthanides. LA lanthanide. That's a fair critique. All right, so we're gonna do a prediction. Sorry, we have a bunch of predictions lined up, and we're gonna drop a bunch of them on you quick because people want to spend their internet points. So let's pop up a prediction right now.
Wait, we don't get to roll these internet points over?
You do, actually. They save. All right, so here's the question. You guys have 2 minutes. How long does it take one molecule? So if you have a fish tank that's 1 meter wide and you drop one molecule on the left side, how long does it take to move to the right side of that fish tank? Is it less than a day or greater than a day? So is it less than a day for that molecule to move 1 meter, or does it take more than a day? This is assuming no stirring, no perturbations. How long does it take a molecule to randomly walk from one side to the other side?
Had to pause to pour a glass.
No worries.
I feel like we have a bunch of scientists here, so
that's what makes it fun.
I thought these questions would be more for like non-scientists.
Can I use binomial theorem? Yes, you can, but you cannot use Google. I don't know if you could even Google this this question. So what is the prediction? Internet points. Spend them now. Does it take less than a day or greater than a day for a molecule in water to move 1 meter?
A typical small molecule with a typical small molecule diffusion coefficient of 10 to the minus fifth centimeters squared per second.
The question is, what temperature is the water?
25 C.
It is standard temperature here. Ask a Scientist Gaming, we follow the rules. Ninja Physics, this might be the one. Get your predictions
1 bar.
in now. All right, Mike, so how long does it take for a molecule to move 1 meter in water?
The diffusion coefficient of 10 to the minus fifth takes 16 years.
16 years. So, significantly greater than 1 day for a molecule to diffuse 1 meter. So the answer there is false. So that's a molecule bouncing around within other molecules. It takes 16 years for it to move 1 meter.
Random walk.
Those of you that said true were not only wrong, but you were like 10 orders or 3 orders of magnitude wrong.
You all need to drink.
Yeah, exactly. You need to drink for getting that one wrong. So congratulations, whoever got this one right. That was pretty even. We had 3 guessing false, 2 guessing true, 70 bet on false, 20 bet on true. So congratulations for those of you that said false. It takes 16 years for a molecule without any perturbation or steering to diffuse for 1 meter. That's pretty crazy.
I have to say, that's why microfluidics, like that's, you know, we have micron-sized channels, so something only has to diffuse a few microns, and so we can take advantage of that. So if it only has to take, if they only have to diffuse a couple microns, it can take seconds instead of a couple years, tens of years. So that's something that, that's the power of going small, right? That's the power of going to these micron-sized channels.
16 years though, that's just mind-blowing. So the factoid I give out whenever I'm asked when I did my stream was, if you create an excited state in the center of the sun through fusion, how long do you think it takes to get out of the sun?
By diffusion?
By energy transfer and collisional motion and all sorts of stuff.
Do we have options, or are we just supposed to guess?
I'm putting you on the spot and making you guess, which is kind of cruel.
Holy
but
thank you. 10,000 years.
You are exactly right. It's 10,000 years to energy transfer. that is amazing, sir.
Is that right?
Yeah, that is right. It's estimated at 10,000 years because the number of energy transfer events plus you have the reverse direction. It's estimated to be 10,000 years for a photon created in the center of the sun to leave the sun's, I don't know, corona and eventually exit to the Earth. So pretty incredible.
That's pretty incredible.
Yeah. All right, we're going to throw another prediction up there. This is a diabetes-related question. Ken, the bonus questions on your exams must be a joy. I don't do bonus questions on my exams. My exams are serious business. You can ask Bronco. All right, we got another prediction up. What % of health expenditures are on diabetes? Is it less than 5% or greater than 5%? Mike, I don't remember. Is this just the U.S.? I think this is just the U.S.
Yeah, I think it's total health expenditures in the U.S. per year.
Yep. So what percentage of total health expenditures per year is spent on diabetes?
Not including health expenditures on fixing my Qbert guy.
That's...
You're going to start playing Qbert pretty soon, aren't you?
Speedrun Qbert?
Speedrun Qbert, round 1, baby.
Oh, man.
Level 1, round 1.
Now I'm going to look. Oh, man. So 4 people think it's greater than 5%, and no one thinks it's less than 5%. I think every time I mention something like that, we get a skeptic just to gamble on the 20-to-1 payout. 10 points at 21 is worth it.
I don't remember the answer. Do you have the answer?
I do have the answer. We have these written down ahead of time. All right. Last chance. Oof. So the speedrun record for Qbert is 35 minutes and 49 seconds.
So there is an end.
Yeah, there is an end. Who
Thank
knew?
God.
All right, so predictions are closed. What percent of total health expenditures are on diabetes? The answer is approximately 10%. So greater than 5%, you take the win on this.
Crazy.
We're at 10% of our health expenditures in the US. I mean, how inflated is that with the cost of our insulin prices and things like that?
It's healthcare in general.
Yeah, that's insane.
Well, that's because diabetes, I mean, you get so many complications, right? You get heart disease and...
Oh, I see. So it's not just the upfront, it's all the implications from it.
Absolutely. All the complications associated with that.
Plus a typical fast food diet. Yes. I mean, that's such a non-trivial amount.
There was a friend of mine is big into like keto diets and things like this. I don't know if you've ever looked into this.
I'm familiar, but not
so
personally.
he showed a plot of like the, you know, when like the food pyramid came out, where, you know, what the food pyramid says, like you should, like the base of the pyramid is like carbs, like bread. About when the time that came out, like then like you see this like explosion in like the weight of the US, the average weight of the US and things. So I don't know, I didn't know if you like subscribe to like keto philosophies and carbless philosophies and things like this. I don't particularly. I think it's too difficult. I think just everything in moderation, I think, is the best way to go. But
yeah. But there was a huge marketing push, right, by, you know, the bread and cereal companies and whatnot. I mean, that food pyramid it's not science-based, as far as I know. No. It's just like a marketing ploy, right? Oh man, Oodles asks, have you guys ever fell for a predatory journal/conference?
I don't think so. I will tell you this. I was invited to give a talk at a conference that was being held in San Diego. So that piqued my interest right there. I love San Diego. And they're like, we'll pay your way. And I was like, okay. Or we'll pay for your registration, essentially. And then I had to pay for the flight and the hotel. It's like, okay, I can do that. I love San Diego and stuff. And so I get there, and it turns out that the conference was held, I don't know, about 20 miles, 30 miles outside of San Diego. Like, it wasn't in San Diego for sure. And it was super expensive conference to register for and stuff. So I feel like that may have been... you could probably classify that as like a predatory conference. But there was people there I knew, so I knew I wasn't just the only one being taken advantage of, at least. okay about that.
So those of you not familiar, so there's something called predatory journals and predatory conferences, where it's basically their goal is to make a profit. And so for the journals, it's not even about peer review. Sometimes they don't even go for peer review. They're just about you pay a certain amount and your article will get published. And so it's kind of pay to play. It's really greasy and fake. I don't know. It's just making profit off of scientific publications, because in a world of publish or perish, you need to get articles out, and sometimes you're willing to pay for them. But we get emails. I don't know how often you get an email, Mike, but I'm at least every other day I get an email from, and the thing is, it's not even to me either. The worst part is it's like to my student who's the first author, like, dear Dr. Wang, you should publish in this journal. I'm like, guys, try a little bit harder. But they're just looking for a paycheck for us to publish in their journal, which is kind of crazy. But yeah, some of them are really elaborate. There was a blog post a few years ago about someone's journey down the academic predatory journal rabbit hole, and like they made up a fake university, a fake website for that university to have the professor.
Yeah, it was University of California Southern or something like that.
University. I'll see if I can find it.
Because I know the guy who did that.
The post?
Yeah, Will Grover. He does microfluidics.
That's awesome, because it was a really
totally, and that's totally his style. He's like just
William Grover. There it is.
Yeah,
extraordinary tale.
It was some kind of California university, right? And they like even took images from a different university and said that it was theirs.
Oh, here it is. All right, for those of you guys that want to read up the elaborateness of someone, what they'll go through in terms of creating a predatory journal. There's the blog post, and it is by Grover, the person you mentioned. Yeah, showing pictures of the website plus the journal. None of this existed. They were just fishing for money. It's crazy.
Well, the problem is, have you heard the other stories where like somebody had a computer, randomly write a paper. So they just took a collection of words and they inserted in random, it was just a hobgob of different words. So they didn't try to make sentences, or maybe they did, I don't know exactly their algorithm, but they essentially published this paper that made no sense. Because they just put in random words and stuff like that, just kind of buzzwords. Essentially, they submitted it to one of these journals that got accepted for publication. So there's like, it didn't even have to make sense, and these journals would be publishing the article. All they care about is getting somebody to submit a paper to them so that they can charge a couple $1,000s to have your publication published. Your manuscript published.
Alright, sorry, we missed a question earlier. How hard would it be to topple the insulin monopoly that's driving inflated prices?
Now?
Yeah.
I don't know much about business. Don't we have a CPA in here?
Is there a CPA in the house? Because it's not just production.
How hard is it to topple any monopoly right now? It's really hard. Right. You got to make an inroad, and then they're just going to buy you out. I mean, pharma right now, it's pretty cutthroat, right? Like, they have so much money, they can come in and just buy out small companies and get their technology and close down the rest of the company or whatever, So it would be hard to compete with that.
Yeah, that's a lot of money. A lot of lawyers. All right, Hamsterlicious
insulin was
oh, go ahead.
Insulin was first produced and they did not patent it, right? So when they first found it out, they did not patent it. so that it could be delivered, so that they could give it to patients and stuff, so that anybody could get it.
So Hamsterlicious has redeemed Take a Drink. So Mike, 10:44 on a Wednesday, drinking. Hope you don't teach tomorrow.
I do not, but I have to go renew my driver's license at like 9 a.m. So.
That's way easier than teaching. All right. Hamsterlicious, thank you for the redemption. We appreciate having an excuse to drink on a Wednesday night while playing video games. So thank you very much for that. All right. So we're gonna pop up another prediction. Whoever wants to bet their imaginary internet points. This one's a personal one, which you may or may not have the answer to or have a good guess on. The question is, how many different gaming systems does Mike Roper own? How many different consoles? We're not going to count PCs in this. This is strictly consoles. Is it greater than 5 or less than 5 consoles does Mike Roper currently have in his house, in his possession, that he could actually play at home if he wanted to?
You've seen my skills.
You did see him last for 48 seconds versus Mike Tyson. Does he have greater than 5 consoles or less than 5 consoles?
You've seen my skills at Qbert for like 3 hours now.
Bet your imaginary internet units right now. Less than 5 or more than 5. And that's one of those things that really comes down to, like, how much do you hoard the things you never use? Because realistically, you play 1 or 2 consoles at a given time, but did you keep the rest of them? That is the question chat needs to answer right now. Does Mike own more than or less than 5 gaming consoles?
So the question is, am I a hoarder or not? I think that's what you just said.
It's part of it. It's part of the strategy. We're doing game theory here and answering this question.
I'm not judging.
That death was unfair.
That was unfair.
All right, 30 seconds less. Does Mike own more than 5 consoles or less than 5 consoles?
How about you, Ken? What do you own? How many do you own?
Oh, man. At least 8.
8?
Yep. Ooh, I'm going to say more than 10, actually. I collect retro games, so that's kind of cheating.
Are you talking about like I was not talking about like handhelds or anything like that.
No. So I have 3 different versions: the Xbox, the Nintendo Wii, the Switch, the NES, SNES, Sega. What else do I have? I have a Nintendo 64. I have a ColecoVision. What else do I have? Yeah, exactly. Do you still have your Intellivision?
I do not.
That's a question I have from you from the bio on the About Us page. How many cans do you have to collect to get an Intellivision?
Dude, did you ever can wrangle?
Yes, absolutely.
Did you?
Yes.
My dad used to make us go out and can wrangle.
Yeah.
And so we'd go out and collect a bunch of aluminum cans and bring them out to the recycling center, and then they would give us money, right?
Yep, they put it on the scale.
That's right, yeah, exactly.
Yep. No, I remember that. Did you actually collect all the money, or did your parents say, like, you worked hard enough, now you earned it?
I think it was more like that. Yeah, I mean, because you'd only get like cents for like turning in the
full garbage bags full of cans, put it on there, and it's like 5 cents.
Well, I'm glad I spent my weekend for 10 cents.
All right, Mike, so what's the answer? How many different gaming consoles do you own right now in your house?
4. I have the Xbox One, Xbox 360, Nintendo, NES, and the Sega.
So 4, less than 5. Oh no, Bronco, is that your first loss of the night?
Bronco is getting a little high on himself, so I think that's good.
He bet on you being a hoarder, so...
Bronco.
That's perfect.
I have lots of issues, that's not one, I guess.
But you don't have your Intellivision anymore.
I don't, unfortunately.
I'm
Did
sure.
you ever play that? It's with the paddle wheel.
Yeah,
It's like a
yeah.
paddle wheel at the bottom of the controller.
Yep, I
And
had
it's
that.
got a numeric keyboard
Yep.
on the
yeah,
top
yep.
of the.
Yep. No, I do remember that. I didn't play it back in the day, but like I started retro collecting like 20 years ago, so I
Okay.
started buying up those guys. But all right, congratulations, those of you that said he has less than 5 consoles. This is going to inspire you to go back and play more Nintendo, though. I'm confident
I
I'm going to see you
definitely-
on the speedrun leaderboard.
Well, if I can figure out the USB controller, gotta get a better USB connection.
yeah.
For some reason, I don't know why these things aren't working. I
Wow,
apologize.
that's crazy, because it was still plugged in. Like, you switched games and it collapsed on you. I don't know.
Right? Because I was playing Tyson, no problem.
All right. So I do have one question that came from Reddit. It's from user Driga Paravukava. What is a beta cell death, and how is it used in research in furthering research into type 1 diabetes? So beta cell death, what is it?
Beta cell death is... So that's usually in type 1 diabetes. That's the autoimmune destruction. So autoimmune means your own immune cells are killing beta cells. Beta cells are the cells in those islets that release insulin. So the immune cells come in, and your own immune cells come in and attack your own beta cells, so you can no longer produce insulin. And that is what is thought to cause type 1 diabetes. So essentially you would no longer be able to produce insulin. That's why kids have to inject themselves, or the parents have to inject the kids with insulin to regulate glucose levels. And this kind of goes into what I was mentioning earlier. They've actually done some studies where they've injected islets from tissue donors into people who have type 1 diabetes, and that can restore their insulin making ability essentially for a few years at least. But the problem is it needs I forget however many, 100,000 of these islets to do that. So it takes several tissue donors to do that, and it's not feasible on a big scale. It's okay to do a small clinical trial and to show that it's feasible to do this, but on a big scale, it's just not feasible. And that's where these other approaches to making islets, like stem cell derived islets, are kind of, their thought is that they could provide a therapeutic approach to type 1 diabetes. I don't want to rage quit on Cuber.
That is next level rage quitting. But you won't be our first rage quit. Aaron Vanucci, a colleague from University of South Carolina, rage quit on it was Madden 2007. I don't remember the game, but interception.
Oh, wasn't
he
it
rage quit.
it wasn't the motorcycle game?
No, nope. It was on football. Yep, PS2. I don't remember which. It was one of the Madden games.
I used to play that a lot too, actually. Were you ever playing I used to love to play the soccer game FIFA.
Yeah.
On PlayStation 2 and stuff.
In person, I promise we will get that going. All right, quick catching up on some questions. Hamsterlicious asked, does it hurt your reputation if you accidentally fall victim to or get published in the predatory journals?
Honestly, yes.
I mean, it depends
Unless
how
it's some
obvious,
elaborate
right?
scheme that you. But like Ken is saying earlier, they address these emails to your student, and it's some of these are pretty obvious. So I guess if you fall for the obvious ones, I would say yes. But if you don't fall to the obvious ones, then probably not.
I mean, with that said, if I'm going to evaluate Mike's publication record, if he has, Lab on a Chip 2 or something like that journal, I wouldn't know if it's predatory or not unless I did my homework, right? So within the community, somebody might know, but slightly outside of it, I don't know.
That's true.
It's hard to tell, because they intentionally pick names that are slightly different than very popular and important journals. And so like Chemistry of Materials versus Materials Chemistry, those are two legit journals, but it's very easy to confuse the two, and you can do it with a predatory journal title. So I don't know, it's kind of hard. If you don't think,
I was assuming that it would be in the same field.
Yeah. No, that's a good question. All right, Omics. How much funding from your grants does Florida State take for administrative costs? I know Hopkins takes like 50% or more.
I would be surprised if Hopkins only took 50%.
Yeah, I was going to say that might be more. Private? Mike, do you want to answer FSU's overhead?
FSU's overhead, I mean, that's a public number. It's 54%.
So
that doesn't come...
Oh, go ahead.
That doesn't come off of our money, right? I feel like I ask for the money that I need to do my research, and then I add in the indirect costs that FSU requires to pay rent and to pay the electricity bills, etc. So I don't see it as taking money away from my research. I see it as just requiring money to pay for facilities and upkeep and things like that.
Yeah, so those of you not familiar, there's something called indirect costs that happens at a university or a national lab. And so when we ask for $100 from an agency, the university will ask for something like 50% overhead. So what you effectively ask for from the agency is $150. 50 goes to the university, 100 goes to you for your research. And that 50 is supposed to go towards things like, waste management and safety issues and facilities and all sorts of overhead. That's what they call it, is overhead things. And so if you're at a national lab, that number is 200% or something like that. If you're at an academic institution, it's typically 50%, at least public institution. I've heard of numbers as high as 65%. And so not only do you need money for your research, but you need money for the university to run normal upkeep of university related things. So yeah, research gets expensive pretty quickly. With that said, they don't take overhead out of tuition, and they don't take overhead out of equipment costs. So if you're going to buy something over $5,000, they won't take 50% off the top of that. But if it's something under $5,000, then they will. Does that about summarize it, Mike?
Yeah, I think so. I mean, again, I think it's just a perspective. Like, I mean, I have a nice lab. And it's air conditioned pretty nicely.
It pays for air conditioning. It's true.
The electricity. I mean, we're in Florida, right? We need air conditioning, and the lights are on all the time. I don't have to pay for water. All these things that you pay for in your house, like, we don't have to pay for in our labs because of the indirect costs. Mm-hmm. So they are getting paid for. It's just that the university has to pay for it.
Yep.
Somebody could make an argument, and I would listen to that argument if the rates are too high.
Yep.
I'm not saying that they're... I don't know. I don't know how they come up with that calculation, but I understand why they need that money. And so on a $200,000 grant, we have to ask for $300,000 because $100,000 of it is going back to the university just to pay for facilities and things like that. So to the general public, I think it seems like a lot, but I mean, the experiments, like Ken was saying, how much does it cost to run a transient experiment? I mean, it's so much money for science.
Yeah, I mean, not just that, like it goes towards, we have shared facilities, like instruments that are housed within chemistry, but not owned by a particular professor, that's owned by essentially the department. And we have full-time staff that maintain those instruments. Like that is absolutely priceless. If you have to troubleshoot a laser not working, it's going to take you months of time and learned expertise, while someone who's an expert can troubleshoot it instantaneously. And so that money goes towards paying those people, maintaining those instruments, so you don't have to. So it is, I mean, if it's too, I agree with Mike, if the overhead's too high, it's just obnoxious, but like, it is priceless to have those resources available to you. I mean, like a machine shop, a glass blower, somebody has to pay for that stuff, and it's not through materials cost and per hour basis, it's through that overhead. So, yeah. I think our 54% overhead is reasonable based on what I've heard from others, but I don't know. All right, we are approaching the 11 o'clock mark. Does anyone have any questions for Mike Roper, analytical chemist, expert in lab on a chip microfluidics? This is your chance to do last minute questions for Mike while he plays keyboard.
Expert in Mike Tyson's Punch-Out.
And an expert in surviving for 48 seconds against Mike Tyson in Mike Tyson's form. Oh, calm down, Bronco. And Scuzzbot came out at the same time. Favorite equation / formula?
Oh, it's got to be the quadratic.
The quadratic.
We had to say the quadratic equation. That was the equation we had to say when
the quadratic.
We thought we had drank too much. If we couldn't say the quadratic, then we knew we had drinking a little too much.
That was your sobriety test? Was the quadratic equation?
That's right. But then you just memorize it really fast. So now when I'm teaching and we have to like solve the quadratic, I'm just like, well, it's obviously -b plus minus squared.
Yeah. Oh man, I don't know if I could do that off the top of my head. I hate teaching that in Gen Chem for the ice tables. I think it's just such a stupid waste of students' time, but that's just me. Like, when the hell are they going to ever have to do an ice table and then solve the quadratic to get an answer? Like, I don't even care if you're a chemistry major.
Maybe they should do it.
Oh, that's step 2? Is it, you have to quote it and then solve for it?
That's right.
I appreciate that. That's way better than the alphabet backwards. There's a quadratic song? Oodles of Omics says there's a quadratic song.
No, there isn't.
No, that can't be real, is it?
Oh, please tell me there is.
I mean, a million monkeys on the internet.
Plus or minus.
We can write it here right now. That is awesome. Learn the quadratic song.
What's your favorite equation?
My favorite equation?
Yeah.
Oh, man. So the one I gave when I was talking, and it's because Eugene hosted me when I was the guest, is the capillary action equation. And it seems like a stupid equation, but the reason I love it is it interfaces intermolecular forces with gravity. And so when you think about quantum mechanics and DFT, it doesn't account for gravity. But when you get to capillary action, it starts to matter. So it's like this threshold where gravity starts to infiltrate into quantum mechanics. And it's this, you need some kind of, unified theory to describe both. And if you can't describe capillary action, you can't actually, describe all of Newtonian plus quantum mechanic physics.
That's cool.
So mine is, it's very altruistic and not applied to my research in any way. But yeah, that's why I like that one. All right, it's 11:02, Mike. You've had a successful run. We did our best with the troubleshooting we had to do at the time, but it's been awesome having you on.
Apologize about that.
That is not your fault. It's the nature of the beast sometimes, so it goes. But it's been a pleasure to have you. Any final words for the audience?
No, it was great. Thanks. I had a blast. It's fun. I'll have to learn more about Twitch and start watching more things.
Well, Mike, your opportunity will be to join us March 31st when Angela Knapp, oceanographer and someone that studies nitrogen cycles in the ocean, and I was also going to play Mario Kart on Nintendo 64. So swing by, join us March 31st. Follow us on Twitter if you guys want to get notifications at least 1 week ahead of time. If you want to catch up on the stream, follow our YouTube channel. We will have the entire stream up live on YouTube in about 2 days because Twitch actually makes me wait 24 hours before posting it. But again, thanks everyone for stopping by. We really appreciate you guys. Questions are awesome. We have a lot of fun doing this, as much fun as hopefully you have watching it. So Mike, thanks again. We'll do it again some point in the future. So thanks again, everyone, and look forward to seeing you in the future.